Start. So welcome everyone to the fourth day of the Sep Research Week. Today we're on Kaurna land. I'd like to acknowledge the Kaurna people, the traditional custodians of these lands. I'd like to pay my respects to their elders, past, present and emerging, and acknowledge the deep spiritual connection that the Kaurna people continue to have with these lands, and acknowledge any other First Nations people in the room or who might be watching online. I'd also like to thank very much Simon Windsor, Constance, the team, and Andrew Burston for putting on such a fantastic week around research. Now, today we're moving into discovery science, and yesterday we heard about what improvement science and development can bring. And Rob Padbury and Grant Phelps and colleagues emphasised that improvement and research can be anyone's business. And I think that's a philosophy that Andrew and others have tried to impart here, that we need to get everyone engaged in research. It's great to see Seaview High School here today. You're very welcome. Thanks for coming. Hope you get something from the day and get inspired to do some research yourselves. Today we're going to focus in the first section on COVID. Now, I think everyone probably knows what that is, and that doesn't need very much introduction. What has been amazing over the past few years of the pandemic is the way in which science has improved outcomes. That might be through careful public health measures, it might be through vaccinations, it might be through treatments, but it's science that has now made it possible to meet in this room and slowly start to take masks off. So locally we've had some really important contributions to the science underpinning the response to COVID. We've had a variety of projects. We've had Anna Ghanasan looking at how to improve masks to stop people getting and transmitting COVID. We've had the work that we'll hear from David and Jingjing around the immune responses and the issues with vaccination. So there's been a lot of work that's gone on locally, and it's great to have two people who have been contributing that work today. Dave Gordon's the head of infectious diseases and microbiology. He's got a long track record of making important contributions to research, both personally and through his work in leading the ethics committee and other endeavours. And he's going to be talking to us shortly, mainly around vaccinations. And then Jingjing Wang's a local researcher, a senior research fellow, who has been really revolutionising our understanding of the antibody responses to diseases and how that can go wrong and how it can produce autoimmune disease, and how occasionally with vaccines antibodies can cause trouble as well. So I'm delighted to welcome both of them to speak today. I'm told that if you log on to the Hoover app, you can get all sorts of information about the week. If you want to vote for the best art, the Discovery Science Artwork Competition, you can vote here. And if you have got some questions that you'd like us to ask the speakers, please go on to Hoover and post an anonymous question. So thanks for coming today. Thanks for joining online. And Dave, over to you.
Thanks a lot, Jonathan. And thanks for the invitation to talk, and welcome to the Seaview High School students. I hope you find this stimulating. So I'm going to talk about sort of two areas. The first part of the talk will be talking about how the mRNA vaccines developed and what work preceded the successful introduction of COVID vaccines. And the second part of the talk will be related to the immune responses, particularly antibody responses to vaccines and natural infection, and try and tie that in with some basic principles of immunology and antibody development. Okay, so when do you think this article was published? I think most of when mRNA vaccines, there was a lot of comments about mRNA vaccines. These are experimental vaccines. We've never used them before. They've just been developed. They've been developed in a rush. Are they safe and are they going to work? And what I'd like to emphasise is that the successful development of the COVID mRNA vaccines was preceded by an enormous amount of work over many decades. So in fact, this paper was published about 2 years before the first cases of COVID, just under 2 years before COVID first appeared. And I don't want to go into this, but basically on the right's just a timetable. I don't want to pull out individual bits, but you can see that there's work going towards the improvement and development of mRNA vaccines for decades. And it was only that previous work that allowed us, we were very fortuitous that this had all happened. And then in fact, some of the major advances and improvements in mRNA vaccines just happened to precede the arrival of COVID by a year or two. And if that work hadn't been done, well, I'm sure we wouldn't be having successful mRNA vaccines. So what's an mRNA vaccine and how does it work and how is it different to other vaccines? A lot of vaccines are protein subunit vaccines where you purify the protein, inject that into the body and you get an immune response. So what the mRNA vaccines do, so the mRNA will encode for the protein of an organism that you think is going to be an important component of the vaccine. And so the mRNA is put into some sort of carrier molecule, injected into the cells or taken up by the cells, and then the protein is translated. So basically then the host's own protein factory takes over to produce that protein that's encoded by the mRNA. So that involves a process called translation of the protein or antigen. And then that antigen in the cell can be released or taken up. It can be presented on the cell surface in conjunction with what's called MHC, class 1 and class 2 molecules, which present an antigen to the immune system, including to B cells. The mRNA is degraded quite quickly, and the mRNA cannot enter the nucleus. So there's no concern about there being any sort of genetic problems or damage to the DNA from having mRNA. In fact, mRNA is quickly degraded. And there was a lot of difficulty in developing mRNA vaccines. And one of the big problems was that the mRNA is very unstable. It was quite reactogenic, and I'll just briefly talk about how mRNA vaccine technology was improved leading up to the COVID pandemic. So as I said, one of the big problems was that the mRNA that you injected into a cell was very unstable, and it wasn't translated to protein very much, so you didn't make very much protein, and so it was very difficult to get much of an immune response. So again, there's a whole lot of alterations and modifications that have been made to stabilise mRNA and to increase the amount of protein that's translated from the mRNA so that you can get a better immune response. So basically these involve changes to the actual sequence. You can engineer the mRNA, change some of the protein codons that make the translation and production of protein more efficient. You can change areas in what's called the 3' untranslated region, which is the bit that's not translated into protein, but still has very important impacts on the stability of the mRNA and how much protein is produced. And you can change other things at the other end of the mRNA, what's called the 5 cap, and you can improve the purification of the mRNA. So these were all really critical components of improving the response to mRNA vaccines. You probably heard or aware that the mRNA vaccines are packaged into a lipid sort of particle. And again, there was decades of work done looking at what was the most effective lipid particles to package the mRNA to allow it to get taken up well by the cell. The other really interesting thing is that the body has systems to recognise foreign RNA. So there are many of the viruses that infect us are RNA viruses, and so there are sensors in cells that can sense foreign RNA. And so one of the major problems with mRNA vaccines was that when you injected the mRNA in, so this is kind of the prototype mRNA that was examined originally. They're these sensing molecules, including things called toll receptors and a whole lot of other things that I don't know very much about. And so what they do is they recognise this as a piece of foreign DNA and they want to get rid of it. So they will destroy the mRNA that's injected so you don't get any protein produced, but also, just as importantly, they cause a lot of reactogenicity and side effects from the mRNA, from foreign mRNA. And it was discovered one of the major advances in mRNA vaccinology was the discovery that if you slightly alter one of the nucleosides, which is one of the components of an mRNA, in particular uridine, and replace the uridine with a related compound called pseudouridine, the body no longer recognises the mRNA that you inject as foreign. So you don't get, lots of systemic reactions and fevers and side effects from the vaccine. So you can see there's been major advances, again, particularly in the last sort of few years, and that was incredibly fortunate for us in developing mRNA vaccines. And that allowed a period of time of just over 60 days between when the sequence of the SARS coronavirus 2 was first reported to the first administration of an mRNA vaccine. So that's just extraordinary and absolutely unprecedented in medicine. But it's on a background of 20 years of going up the wrong track and originally very little, if any, enthusiasm for mRNA vaccines. If you're interested in the story of the mRNA vaccines, there's a great paper here in Nature recently, which also brings out all the sort of personalities, which are always interesting in these sorts of things. That's, who's going to get the Nobel Prize for, which were the key discoveries in these mRNA vaccines. Okay, so I'm moving already to the second part of the talk, which is really to just tell you a bit more about how effective are the COVID vaccines, what are they good at, where do they fall down, what immunological principles might they sort of demonstrate. So this is, as you all know, both the Pfizer and the Moderna. I should mention the Moderna. Moderna is modified RNA. So Moderna discovered the uridine, pseudouridine substitution that appeared to be critical. So this is the Moderna vaccine, but the results were virtually identical. So basically, this shows you that this is protection against COVID, and so if you had the vaccine, you get very, very little COVID infection, and the placebo group got a lot of infection, and so the overall vaccine effectiveness was about 95%, 90 to 95%. The FDA had originally said if you can get 50% effectiveness of your vaccine, that'll be great, and we'll take that. So I think probably everyone was very, very surprised, I guess, about getting 90 to 95% protection. and, you know, even higher protection against severe disease. So in this study, there's over 98% protection against severe COVID. So that looked, you know, this is a year or so, or 18 months or so ago, so, you know, it looked pretty promising that, you know, maybe COVID wouldn't hang around too long. But unfortunately, the virus has got a great ability to mutate. It's an RNA virus, and so the replication of RNA viruses by an enzyme called RNA polymerase is relatively it's error prone, and it has a relatively high mutation rate, probably about 1 in 5,000, 1 in 10,000 mutation rate, which is much more frequent than DNA replication. And so what people suddenly noticed, this is a study in vaccinated workers and unvaccinated healthcare workers, and, you know, the vaccine was obviously very, very effective until sort of middle of last year, and at that point in time you can see there was a big resurgence of cases in both vaccinated and unvaccinated. It looks like there's no effect of the vaccine here, but it's biased a little bit because most of the workers were vaccinated, so these are not, the relative risks were still lower in this group. And you can see this coincided with the appearance of the Delta strain of the virus. So this raised all sorts of questions, you know, does the immunity improve or wane over time? What's the effect of boosters? How well, you remember that all the mRNA or all the COVID vaccines are based on the Wuhan strain original sequence. So these subsequent strains are all variants, and although they're obviously at least partly related to the Wuhan sequence, they are different variants. So, you know, how well are their variants covered, and is there protection against serious illness and death? Is that sustained? So this shows you the sort of protection over time against mild and more severe illness. And this is the AstraZeneca vaccine, which I won't worry. I'll just talk about the mRNA vaccines. So what this is showing, this is the Alpha variant, which was one of the early ancestral strains, and the Delta. And so you can see over time that the protection against symptomatic disease just drops off a bit. Or probably drops off about 20% perhaps over 6 months or so. But the protection against hospitalisation is pretty well maintained, and the protection against death is pretty well maintained. So although the vaccines start to lose efficacy against getting the disease, they still hold up very well in protecting you against severe disease. Although the protection against more severe disease does fall off a little bit in the higher risk population. So again, this is a similar graph to the previous one, but you can see now that this does drop off a little bit more quickly in people who've got other risk factors and more clinically vulnerable. Again, but it holds up pretty well still in the clinically in those that aren't clinically vulnerable. So there's not much drop off. So there's sort of some good and bad news at this point in time in terms of the vaccines. So why is this immunity waning? And is there anything that we can do about it? So this is a study that was done at Flinders with immunology, infectious diseases, and also in conjunction with the Doherty Institute, and it looked at a series of patients looking at the decay of their antibody over time. And you can see that there's a gradual drop-off. It appears to be a sort of two-component delay. This is probably losing IgM. And Jingjing will talk a lot more about this, but the immunology and proteomics group here have established a method for actually sequencing the individual COVID antibodies that develop and tracking an individual clone. And the significance of that you'll see in Jingjing's work where she's identified a particular clone. So just quickly, because I don't have too much time, so how about a booster dose? So the booster dose is definitely effective at protecting you against severe disease. That's fairly clear cut. And I'll just skip that for time. But so I'll come a bit more now, just to the last little bit about cross-protection against different vaccines and the sort of immune response that you get. So this is a bit of a complicated slide, but this is measuring the amount of antibody, or what's called neutralising antibody. That's the antibody that will stop the virus growing. And so if you have natural infection, you produce antibody against the sort of ancestral strains and reasonable antibody against Delta, but you can see very little antibody against Omicron. If you have 2 doses of vaccine, you still don't get much Omicron protection. But if you have 3 doses of vaccines, you do start to get Omicron protection. So that's why it's so critical that we get 3 doses, at least 3 doses of vaccine. And at the moment we have 25% of our population who have not had a 3rd dose and are not going to get good protection. The other interesting thing that I'll just draw your attention to is that the response to the booster dose is much broader than originally. So the booster dose is still against the ancestral virus, but you start to get antibodies produced against all these Omicron. These are all Omicron variants, all these BA1s and 2s and 4s and 5s and things you've probably heard. So you can see with 2 doses, this is the protective or approximate protective level. You can see with 2 doses, you don't get really Omicron, you don't get any coverage against these. But the 3rd dose gives you much broader neutralising antibodies. This is a bit of a summary slide. It's just another paper, but I don't have time to show the data. But it's really interesting because it shows us what happens in response to various scenarios. So, and the breadth of the immune response. So if you're unvaccinated and you get infected with the ancestral, these early strains, you get a good antibody response against those early strains, but you don't get much in the way of an antibody response against Omicron. If you're unvaccinated and get infected with Omicron, it's really interesting in that the immune response is really narrow. And so you really only get a BA1 response, and you don't get antibody in response to these ancestral strains. Whereas, as I've shown before, with the 3 doses, you do get a broader response. And if you get vaccinated and infected, which is probably lots of us now fall into that category, you get the greatest response and you get the broadest response. So I know I'm short on time, so I'll just try and wrap this up in terms of the immunological principles, what's happening here. So when the body recognises a foreign antigen, amongst other things, that antigen is recognised by B cells that have an appropriate B cell receptor that can detect that antigen. And that original B cell can produce antibody that's, this is what's called a naive B cell, and so it will produce antibody that's part of our sort of genetic background sequence, although the process of making that's quite complicated. And then, so that will produce the B cells will develop into plasma cells and they'll produce antibody. And many of those plasma cells will just die off relatively quickly. Some of the B cells enter the lymph nodes in particular and what's called the germinal centres of the lymph nodes, and then they can mature, their antibodies can mature and can develop and can improve by a process called somatic hypermutation. And so basically you then make what's called long-lasting plasma cells and memory B cells, and gradually the B cell population that's in there, the ones that produce pretty poor antibodies die off, and the ones that produce really good antibodies turn into permanent, long-lasting plasma cells, which may be in the circulation, just continue to produce low levels of antibody all the time. So plasma cells in the germinal centres, or memory B cells, which are often in the blood, don't produce any antibody, but those memory B cells, when they're exposed again to the infection, they're all there, they're already there, and then they can produce these much better antigens, as antibodies, than the original antibodies that were made. So I don't have time to talk about the cross thing, but just in summary, so the development and effectiveness of the mRNA vaccines has exceeded all our expectations, I think. The immunity wanes, but protection against severe infection holds up well, but we must get a third dose at least. So the booster doses enhance and broaden the responses, because you've now the booster responses are occurring on a background of already existing somatically hypermutated antibodies that are already primed to be better. So you can get reasonable cross protection. The natural infection with Omicron, if you haven't been vaccinated, is very, very narrow. So if these ancestral strains come back again, maybe these people won't have very good cover. And I think it's obvious that we need a detailed characterisation of antibody responses. Is critical to sort out what we do with new vaccines. And as you know, there's a vaccine about to be released in Australia that's a mixture of the ancestral antigen and the Omicron, but it's the Omicron BA1. Yep. So I'll finish up there because happy to answer any questions.
Dave, thanks very much indeed for taking us through that. Invite some questions from the audience and on the Hoover app. I might just start. So you emphasised that the vaccines can stimulate, some angry immune response. I guess trying to hit the sweet spot. You want a vaccine to excite the immune system, but you don't want it to get too angry. Yeah. How do you achieve that middle way?
I mean, one of the part of the angry immune response is because it's recognised as foreign, and so the systemic responses to the original mRNA vaccines were quite strong. Everyone just got terrible fevers, but I mean, it is, as you say, it's a bit of a sort of fine line. But the problem is that immune response destroyed the mRNA as well, so you didn't get much protein translated. Yeah. So you don't want an angry immune response that destroys the mRNA, because it's not an immune response that's in response to the antigen, it's to something else. Yep. It's never going to help you. But, there'll always be some immune response to the foreign antigen. And there's a degree of correlation between the severity of this kind of reactogenicity and the degree of antibody response, although it's not absolute.
Okay. Yeah. Any other questions at all? Andrew?
Look, thanks, Dave. They've been going slightly sideways. There's been a whole bunch of areas that have been pretty hard to crack, and I think we are cracking a vaccine for malaria now. But, what's the future with mRNA vaccines? I guess it's the first question. Are we going to be able to start development of some of these very complex problems that we've not solved in the past? And on the other side of that equation, we're now hearing that we've got mucosal vaccines coming, which might be longer lasting and more specific. So can you just take us through that, that little conundrum?
Yeah, vaccines against respiratory viruses have always been, incredibly difficult. There really hasn't been much success with respiratory virus vaccines. So I think, the success of COVID there's a whole lot of other vaccines that are now sort of under development and under testing. I'm involved with a Moderna influenza vaccine, RSV vaccines, so respiratory virus vaccines, but others. And also there are cancer vaccines, mRNA cancer vaccine. In fact, a lot of the early development, they didn't think they were going to be much use for infectious diseases vaccines, but they might be cancer vaccines. So the second question about the COVID vaccines.
Just really, could you just give a bit of a crystal ball and tell us?
Yeah.
Just crystal ball and tell us a little bit about what's going to happen with the mucosal vaccines, because we've been told to prepare for a pandemic every 6 months. That's one of the naysayers out there, and we'd like to know we're going to do better.
Yeah. I mean, I suppose we did pretty well, really, when you actually think about it, but that's the only thing that really got us through. But the advantage of mRNA vaccines is you can make them and change them, in a couple of days. It's not hard to do that. I didn't show the data on the hybrid vaccine, which is half BA1 and half ancestral strain, but that gives somewhat better responses against Omicron and better cross-protection, broader protection as well. Although it's a BA1, there's good BA5. Now, look, what's going to happen in the future, who knows? We haven't had a major new variant since Omicron, and Omicron's almost 12 months old. Not quite, but it's getting up to 12 months. So there's obviously a degree of population, protection, at least against severe disease. So look, I think it's really difficult to know. I'm sort of optimistic that there won't be too much major changes. It would be a bit sort of catastrophic if there's a major immune escape strain. I think that's pretty unlikely.
Question there, please.
How are RNA vaccines adapted to new variants? Because you did say how the original or ancestral variants they came from Wuhan and the first vaccines were made with that, but how did you adapt them to the newer variants?
Yeah, okay, no, that's a good question. So the new vaccine that's coming out is just based on the spike protein, which is the part of the virus that attaches to cells, and so if you can make antibodies that block that attachment, that's basically how the antibody component of the vaccine works. So, as I suggested, there's a degree of cross-protection across the different strains, but it varies with the different strains, and even within Omicron it varies a little bit between, BA1 and 2 and 3 and 4. But there's nothing to stop you updating your mRNA vaccine quite quickly. It might be better, ideally we probably would put a BA5 spike protein in the mRNA vaccine now rather than the BA1, but in data I didn't have time to show you, but as I said, a BA1 vaccine also gives you an improved immune response against BA5. So again, it relates to this sort of idea that we can get a broader immune response, so we can get protection against different strains, even though the vaccine strain and the vaccine spike protein is a bit different to these other ones, as the immune response and antibody response can mature and develop.
All right, look, we might have to move on there, but Dave, thanks very much for your leadership of infectious diseases through this time. I know it's been incredibly powerful to have your leadership and enable us to cope with COVID in a really effective way. So just next, Jingjing, please come and tell us about some more detail around the antibody responses to COVID.
Good afternoon. Today I will talk about a dangerous autoantibody in vaccine-induced thrombotic thrombocytopenia, also called as VIT. So what is VIT? If you can think back to the beginning of 2021, we were in a race against the virus by immunizing the world with a COVID vaccine. As a larger number of the population got immunized, we realized there was a very rare but life-threatening blood clotting reaction linked to Johnson & Johnson and AstraZeneca vaccine. So what do we know about VIT? VIT mainly occurs in people who received adenovirus vectored COVID-19 vaccines, but not mRNA vaccines. The incidence varies from 1 in 2,500 to 1 in 50,000. And there's no clear risk factor, but more common in young females. It occurs 1 to 2 weeks after the first vaccine dose, suggesting an early priming event. Thrombosis often occurs in unusual sites in the body, notably sinus veins of the brain. And you must remember, VIT has led to the restriction of AstraZeneca rollout in Australia in 2021. Interestingly, VIT presents striking clinical similarity to HIT, that is, heparin-induced thrombocytopenia. HIT is an important, clinically important adverse drug reaction, and it is induced by antibodies specific for complexes of platelet 4, the positively charged, and the polyanion heparin. And anti-PF4 and heparin antibodies can activate platelet via their Fc receptor, and then trigger blood clotting events combined with platelet consumption. Similarly, in VIT, anti-PF4 can activate the platelet via their Fc receptor. And the end result is the syndrome of thrombocytopenia caused by platelet consumption and immunothrombosis, in which anti-PF4 antibodies are common to HIT and VIT. However, binding of VIT-associated PF4 antibodies to PF4 and platelet activation are both heparin-independent. Extensive research has been carried out to understand VIT. It has shown that VIT is mediated by antibodies against PF4. In a key advance, Nazi's group has mapped the binding site to a single conformational epitope, which is located in the heparin binding site and distinct from the epitope bound by the serum from HIT patients. In other words, VIT antibodies and HIT antibodies actually bind to very different locations on PF4. From this front view, it is easier to see. So the red dot, like north and south poles, are heparin-dependent antigen sites, and the blue ones represent heparin binding site. And this region is rich in arginine and other lysine residues, so very positive. Research also showed that VIT is not related to spike protein. And also, VIT antibodies do not cross-react with the spike proteins. And interestingly, most of pathogenic anti-PF4s in VIT are transient, despite of a high persistent binding levels. Given their causal role in VIT, identification of the molecular composition of these anti-PF4 antibodies and their antigenic targets is crucial for understanding of the pathogenesis and for developing better diagnostics and treatments. Based on the fact that anti-PF4 binds to a unique and shared epitope on PF4, we hypothesize that VIT patients mount an identical powerful antibody response to the PF4 target. We believe better understanding anti-PF4 genetics and structure will identify interventions to mitigate the risk. As we know, antibody has a basic Y shape, 2 identical heavy chains and 2 identical light chains. and the VDJ regions are found in the heavy chain variable region. In general, heavy chain CDR3 is this region, is the major site of antigen binding and defines the clonal type of the antibody. Human IgH locus is located on chromosome 14, and kappa is on chromosome 2, and lambda is on chromosome 22. For IgHV, there are around 58 functional genes, up to around 19 reported alleles per gene. Studies at B-cell level has been carried out to analyze clonality and IgV gene usages of autoreactive B cells, but to my knowledge, no studies have been done for the B-cell repertoires in VIT. More importantly, serum antibody repertoire, the final product of the humoral immunity, has remained largely unknown. Currently, conventional binding assays, such as ELISA, are used to detecting overall binding activity, but they are unable to pinpoint the pathogenic clones in the complex VIT patient serum. Here, I would like to show you our innovative way to profile serum anti-PL4 and their clonalities by using mass spectrometry sequencing. So, mass spectrometry is a powerful analytical tool to determine amino acid sequence of a peptide by measuring their mass-to-charge ratio. And Flinders Immunology Proteomics Group was the first in the world to use this technology for sequencing human autoantibodies in complex serum from autoimmune patients. There are challenges in bioinformatics. In other words, how to analyze or interpret the huge amount of sequencing data. Antibodies are very complex in human serum, and they are extremely similar in their sequences, as you can see from these slides. So how to overcome this? We developed in-house program to distinguish among autoantibody clones, and we also use a powerful search engine called Pixel Studio for data analysis. This technology also requires database for matching the sequences. However, unlike cellular proteins, there is a limited database available for antibody repertoires, especially for the diversity region, which is the CDR region. To solve this, we use paired BCR sequencing as a reference database. We also developed a difficult technique called de novo sequencing. Importantly, we use extremely high-end mass spectrometer to sequence intact clonotypic HCDR3 peptides. Here shows our proteomic platform. It consists of two parts. They are discovery proteomics and quantitative proteomics. Briefly, anti-PL4 antibodies are purified from VIT serum by using PL4-coated magnetic beads, are digested with enzymes, and subject to LC-MS. The clonotypic peptides are identified by de novo sequencing, and these peptides will be used for relative quantitative proteomics for tracking in the longitudinal samples. In this study, 5 patients with VIT were recruited by our collaborator, Dr. Chi-Vi Tan. He is a hematologist in Ra, and he is also the president of Thrombosis and Hemostasis Society of Australia and New Zealand. As you can see, the patients here present very typical clinical features of VIT, including decreased platelets, and also elevated D-dimers. So this is the most exciting bit. So mass spectrometric sequencing of anti-PF4 antibodies revealed that a single heavy chain species paired with a single light chain in all 5 patients. As I mentioned earlier, HCDR3 peptides are the clonotypic markers of serum antibodies. Here, strikingly, here we identified stereotypic features characterized by exactly same, like identical HCDR3 length and homologous sequences, together with shared binding motif. Interestingly, we searched the E-receptor database for any CDR sequences with this kind of a motif. Surprisingly, we only found 23 out of more than 30,000 sequences showed matching to the conserved motif. So suggesting this clonotype antibody actually is very unique in wheat antibodies. Another remarkable finding is that all light chains were encoded by IGLV 32102 and showed exactly same LCDR3 length with a shared amino acid motif. So we have sequenced many autoantibodies in our group, and we found it is very unusual to express exactly same light chains in all unrelated patients. So that makes us think, is this IGLV 32102 light chain a unique fingerprint of wheat antibodies? This table shows the alignment of different IGLV 321 alleles, O1, O2, and O3. Notably, O2 allele, which was found in our antibodies, expresses an acidic and also like a negative charged DDSD motif, suggesting the potential importance in antibody binding to the positively charged PF4 epitope. The frequencies of different alleles vary among ethnic groups, with the highest prevalence of O2 allele in European people and the lowest in East Asian. The same group also reported that O2 allele light chains represents around 4% of a lambda chain transcript from PBMCs in healthy donors. This light chain has also been found as a very minor component of the total serum repertoire. Taken together, the dominant expression of IGLV 32102 combined with the very unique HCDR3 signatures can be regarded as a unique fingerprint of wheat antibodies. So to understand how this polymorphism impacted the antibody structure, we used our MS data sequencing data and through a series of computational tools. And the most important one is a deep learning guided modeling of CDR region conformation. And then we used a flexible docking algorithm called HiDoc to dock the modeled antibody into the likely binding orientation on PF4. And our PhD student, Alex, has set it up nicely. So this is the fly-through of one of our wheat antibodies' interaction with the PF4. So as you can see, it is quite amazingly modeled. So As you can see from the model, showing HCDR3 is the central role, like spiking the heparin binding site, and also the LCDR2 region, DDSD motif, is predicted to interact with residues on PL4. And you can see HCDR1 and ICDR2 also involved in interaction with PL4. Our study has attracted quite a lot of attention. For example, Root has published a commentary, and the study was published in Blood, and also it was highlighted in a recent international conference. So here I would like to show you the model on generation of a dangerous anti-PL4 clonotypic autoantibodies. Firstly, pre-existing germline precursor clone with IGLV32102, and usually these clones will be like a very low affinity. An exposure of a PL4 epitope by adenovirus vector, and then undergoing PL4-driven affinity maturation and clonal expansion. And usually those ones will be high affinity antibodies. And this anti-PL4 will activate platelet and trigger VIT. So in summary, marked clonotypic stereotypic consistent with identical pathways of pathogenic anti-PL4 production in VIT, involving genetic hardwiring and antigen-driven selection. And this was the first serum cloning of a dangerous human autoantibody. So what comes next? First, longitudinal studies using quantitative proteomics with personalized molecular barcodes that we identified in our previous study. As I mentioned earlier, several studies have shown that anti-PL4 antibodies remained persistently high level on conventional assays. Unexpectedly, the functional activity of these serum antibodies dramatically declined over time. So to understand this, we will investigate the evolution of pathogenic anti-PL4 at a molecular level, so which is invisible on conventional immunoassays. To unravel the molecular pathway of the functional deactivation of these pro-thrombotic antibodies. Second, reverse engineer recombinant anti-PL4 antibodies for personalized diagnostic and therapeutics. To achieve this, we have collaborated with Janssen, and they are making some monoclonal anti-PL4 antibodies this moment, just based on our amino acid sequence from our study. So these monoclonal antibodies will also be used for cryo-EM to establish the structural basis of antibody and antigen binding. The last but not the least, genetic screening of Ig genes to identify at-risk subjects. Here I would like to thank our Flinders immunology team and also Flinders proteomics, especially my mentor, Professor Tom Gordon, and also our research scientist, Brady Armour, and the PhD student, Alex. And special thanks to Professor Tim Chatway and Dr. Alex Colella, and also other collaborators from RA and Calgary Hospital and Janssen team for our future work, or actually the work this moment. Thank you very much.
Jingjing, that's great. Thanks very much. Astonishingly precise molecular work. So I can't just go to any lab and just ask for them to sequence an antibody then. This is a bit more complicated, is it?
Well, yeah, I believe not, because I think we have very, very specialized skills and expertise, and also like we are supported by the proteomics facility. So I don't believe anyone can do that.
Yeah. So you mentioned the importance of VIT and the overlap with HIT. So do you know whether these stereotypical antibody responses underlie HIT, the heparin-induced thrombocytopenia? What's the commonality between the PF4 antibodies there?
Okay, so as I presented in my slide, actually HIT antibodies and VIT antibodies, they bind to very different epitopes on PF4. And also we already sequenced some antibodies from HIT patients. So even though they present very similar clinical symptoms, but actually the antibody repertoires are very different. And also there are differences about, like, firstly, the binding site are different, and also the duration of persistency of the antibody is different. So in VIT, anti-PL4 can be persistent high for like up to 7 months, but in HIT actually only lasts for 50 to 80 days. So they are quite different.
Good, thank you. Some questions from the audience or online? Tom.
Yeah, thanks. We can actually get a signature within 48 to 72 hours, and we're offering that to the world, you know, but the world isn't very interested right now. The paper's been in Blood, we had one citation. It's just such revolutionary work. It's completely turned immunology upside down, and the amazing thing is that from a small amount of patient serum, we can reverse engineer and make a monoclonal antibody. And that's absolutely revolutionary. But again, no one's interested yet. But, you know, with VIT, everyone holds on to their sera. They won't exchange it. Even in Australia, interstate, we can't get anything. Even though we know they've got, 30 or 40 patients. So it's a really strange thing. When you've got a new disease, whether you're from America, Europe, Australia, there's no cooperation. Fascinating, isn't it?
Well, let's hope that someone's watching today that'll be enticed to collaborate better.
Yeah, thanks, Junjing. You implied that you needed some antibody maturation and mutation in the pathogenesis of VIT, but the onset of the disease is quite rapid after the vaccination, and I wouldn't have thought there would be time for too much antibody maturation to occur over that time. So is the germline antibody sequence enough to cause VIT, or do you need some antibody maturation or affinity maturation or mutation?
That's a really good question. Actually, there's a similarity in VIT and HIT, because in VIT, actually the antibodies also happen really fast, even within 1 week. For example, after cardio surgery, and they had a heparin treatment, and that they will develop a HIT within 1 week, but the predominant IG class is IgG, so there's no IgM, and same for VIT. So it happens really fast after first dose. So some explanations saying that's actually there's already pre-existing anti-PL4 antibodies in the body. So actually the vaccine or the heparin actually is just the stimulated and all, and so that's not really a primary immune response. So that is kind of innate.
Any other questions? Andrew.
Thanks, Jingjing. And in HIT, I think you mentioned 56 days, which is about right, because if you have heparin-induced thrombocytopenia, and you wait about 3 months, you can give the heparin again and you don't usually get the response. So 2 questions. Why is that? It suggests that there's something else going on, another factor. And is that likely to be the same in VIT? In other words, if somebody's had VIT once, would they get it again, or could you now presumably give it safely? So what's the other factor, or have you any ideas how we would understand that?
Yeah, so actually there are already some studies published, just like for VIT patients, and once they receive second dose of AstraZeneca or Pfizer, there's no recurrence. They don't happen again. So the mechanism is not really fully understood, but one possibility is anti-idiopathic antibodies. So just like once you got anti-PL4 antibodies in the body, and when that like antigen happened again, but probably there's already like the body already mounted enough anti-idiopathic antibody to neutralize that. So probably that is why it didn't really happen again, because that has been already reported like in New England Journal paper. So they had around 60 patients. They had a VIT, initially VIT, but then they either receive AstraZeneca or Pfizer or Moderna, but none of them happen again. So I think, even though it is not fully understood, but I think probably anti-idiopathic antibody is one of the explanation.
All right, look, we're a bit tight on time, so we might close there. But please join me in thanking Jingjing and David for the great presentation.
Thank you.
I guess we might start. So good afternoon to everyone. My name is Sara Tommasi. I'm a medical scientist in clinical pharmacology in the Division of Medicine, Cardiac Clinical Care, and I'm really honored to chair this session today. I would like to welcome you all to the Discovery Science Day free paper presentation session, and I will soon proceed with introducing our speakers. But first, I would like to acknowledge that the Ghana people are the traditional custodians of the Adelaide Plains, the land on which we gather today, and pay respects to the elders past, present, and future. We recognize and respect their cultural heritage, belief, and relationship with the land. We acknowledge that they are of continuing importance to the Ghana people living today. So just to briefly recap the rules of this session and I guess a few housekeeping information. The session will be attended by a live and virtual audience and will be live streamed and recorded. Each presentation is up to 10 minutes, and after all 5 presentations have been delivered, there will be a joint Q&A session of about 20 to 30 minutes. We encourage our online audience to submit their questions using VUVA, while Tanya from the fantastic research office team will run a microphone among the participants in the room. I would encourage the attendees to specify the speakers the question is addressed to and to avoid the use of jargons and abbreviations when possible. We will try to keep the time of the session as close as possible, and the speakers will be alerted when there is 1 minute left to their presentation. The Flinders Foundation has generously offered to sponsor the awards for the Best Discovery Science free paper presentation and the Best Discovery Science poster. And we encourage all of you to express their vote by answering the polls on VUVA. So, yeah, and if you want to play around a little bit with the app, there's a lot of information that can be gathered, a space for questions, there's an art competition that you can vote on, and a lot of things that can be done. So, without further ado, I would like to introduce our first speaker, which is Dr. James Chan. Dr. Chan is a research scientist whose focus is on atherosclerotic plaques. And the title of Dr. Chan's presentation is Collagen Composition in Endarterectomy Atheroma Plaque, a Determinant of Plaque Stability. Thank you.
Good afternoon, and thank you to everyone. Thank you to the chairperson and everyone attending here and online. Okay, I'll be presenting my preliminary findings on the collagen composition of endarterectomy atheroma plaque, a determinant for plaque stability. I will briefly take you through the introduction of arteriosclerosis, my objectives, my methodology, results and discussions, and conclusion, and finally, future work. So, a brief introduction of arteriosclerosis. It is one of the leading causes of death in Australia, and in 2019, it contributes to 25% of the total death. And it's defined by the thickening and hardening of the artery walls due to the buildup of plaque in the inner lining of an artery. And the risk factors are unhealthy dietary habits, hypertension, obesity, smoking, and dyslipidemia. In general, arteriosclerosis can be divided into 2 types, the stable and the unstable plaque. The stable plaque are actually defined by the hard calcified overlying endothelium, which is strengthened with a thick fibrous cap. And the unstable plaque are usually quite thin, fibrous cap with a thin fibrous cap, more vulnerable to rupture. And once it ruptures, it will release the thrombogenic substance and resulting in stroke. So my objective in this experiment is to determine the plaque stability if it's directly proportional to the collagen content, to quantify collagen content in atheroma plaque using a trichome stain and also an open soft software such as ImageFiji, and also to compare the collagen content difference between the lesion versus the non-lesions area. So in this slide, we can see the atheroma plaque is usually collected during endarterectomy procedure. And on the top here, you can see the one on my left is actually the fresh atheroma plaque with the tunica media showing. And the one on my right is actually the tunica intima. And the picture at the bottom is actually a formalin fixed paraffin embedded process H&E section. So in the next slide, we'll zoom into the paraffin section, and I can show you the morphology characteristic of the atheroma plaque. So in this picture, where the label C actually are the calcified area of the atheroma plaque, and it's the mineralization of the collagen and other substances that we are still trying to find answers. And besides calcification, we also have the adipose tissues. And in here, if we further zoom out in the little yellow square, we can see these are the adipose cells. And we have the angiogenesis in the plaque itself. And another common characteristic of the atheroma plaque is the deposition of the cholesterol crystal, as seen in these pictures. And these are the cholesterol crystals that you can see in H&E stained atheroma plaque. So to take you through the methodology, this is the same previously stained H&E section, and it will be stained using a trichrome stain, as the name implies, it consists of 3 colors. The principle of the stain is actually by size displacement of the dye, and the picture on this to the right is actually the meson trichrome stained atheroma plaque. So the collagen will be stained green, muscles and red blood cells will be stained red, and the nucleus will be stained dark blue to black. Then the stain section will then be scanned using the Olympus VS200 image scanners at 10 times magnification, and the images are then measured using Image Fiji, a open source software, and a macro using published by Kennedy et al. And this collagen content is compared with the cap area. So in this picture, we can see a good cap area, and this is the cap of the atheroma, and this is the necrotic core. And on this picture as well, we can see a segmented atheroma cap area, and this is one of the caps. It's been segmented out and took for measurements, and this is another cap here as well. Here itself, we can see content underneath the cap, and this is the internal lining of the tunica intima. So overall, in total, we have 9 samples that were measured, and some of these samples, we have multiple sections. And from here, we can see that out of the 9 samples, all of them have shown increase in collagen content, except for one. And we postulate that that one sample that has shown a decreased amount actually could be in the early stage of the disease, or possibly in a process of destabilizing. So it might become an unstable plaque. So from the results that we showed, so the increase of collagen is approximately from as little as 2% to more than 100% increase. And multiple reports have suggested that the amount of collagen that actually increase is actually inferring plaque stability. And also, another function of collagen is actually to has a dynamic contribution in regulation of arteriogenesis. In my personal experience in using Image Fiji, I find that it is actually quite a useful software for biological imaging, and it can also be automated with a combination of a software of a script. And the only drawbacks that I face is actually when I process larger files, it tends to be quite slow and also running out of memory. And this could possibly due to the desktop that I am using. So in conclusion, I hope that I have managed to show you that meson trigram and image VG are useful to measurement of collagen in atheroma plaque, and also the higher content of collagen is present in the fibrous cap, and thus inferring plaque stability. So in our group, my future work will be focusing on examining the role of collagen in remodeling and mineralization. And also we have an interest to examine the gut dysbiosis effect on atherosclerosis deposition. And I'd like to thank everyone for their help. Thank you.
Really great start. Thank you, Dr. Chen, for this presentation. And let's now move to Dr. Vincent Wong, a postdoctoral researcher in the Renal Research Lab. And he will present on single nucleus transcriptome profiling of kidneys undergoing compensatory growth. Thank you.
Hello, good afternoon, everyone. Thank you for giving this opportunity to share some of my preliminary findings. My name is Vincent. I'm working in the kidney lab here in Salon. So today I'll be talking to you about how we use this exciting new technology using this single cell to better understand kidney compensatory growth. So chronic kidney disease is a very common disease where there is a gradual loss of kidney function and size over time. So it is estimated that there is 1.7 million Australians that are living with early sign of kidney diseases, but they are not aware of it. So when the kidneys no longer function adequately, people usually require dialysis or a kidney replacement from a healthy donor in order to survive. So therefore, there is a need for a better treatment or to boost the kidney functions. So often when a healthy donor donates a kidney or people undergoing kidney removal due to cancer, it's fascinating that their remaining kidney enlarges and improves in function to compensate for the loss of the other kidney. So, and no one knows how it works. This process is actually called compensatory renal hypertrophy. And our lab is it's interesting that like people with just one kidney, they are able to survive up to 50 years, live a normal healthy life, partly because of this growth in the remaining kidney that sort of do the load of what is meant to be a load of 2 kidneys. So our lab is particularly interested in how this process works, and we use certain molecular techniques and which I'm going to talk about, the single cell, to trying to better understand this mechanism of causing this kidney hypertrophic growth. We are hoping that with this finding, we are able to use it to better promote kidney healthy growth. So a postdoc in our lab, Darling, to be able to look at this, a postdoc in our lab, Darling, she has set up this compensatory kidney growth mice model where she performed unilateral nephrectomy to remove a kidney and have a sham control where both kidneys are intact. So the first question we want to ask is, do we see kidney growth in the remaining kidney? So yes, we do see it. So in 2 weeks' time, we are able to see that there's about 25 to 30% increase in the kidney weight to body ratio. So the next thing we're trying to do is to collect kidneys from these 3 time points, the 24, 48, and 72 hours, to try to look for the early signature or early trigger for this compensatory growth process. So we mince up the kidney tissue and perform bulk RNA sequencing where we measure the gene in the kidney to try to look for differences between nephrectomy and sham operation mice. So when we look across time point, we saw a substantial change in the number of genes that were switched on and off from 24 hours to 48 hours. What we're trying to do is trying to relate the change in genes to what it means. So we carried out a pathway enrichment analysis. And here we are able to see that there are unique signatures that's associated with each time point, and that's 24, 40, and 72 hours. So here in the 24 hours, we see an increase in mTOR signaling, in cholesterol signature, whereas in the 48 hours we see some increase in mitochondrial transcript, and 72 hours there's some cell cycle changes. So the cholesterol signature that we are seeing at 24 hours hasn't been reported, and it's quite novel. And so we want to really delve deeper in looking at what genes is involved in this cholesterol signature. So here's the cholesterol pathway showing all the enzymes and genes that are involved in making cholesterol. So we're trying to look at what are the genes that changes. We saw not just one gene being switched on, but multiple genes in the cholesterol synthesis pathway that is actually switched on. So which suggests a really strong significance of how important this cholesterol pathway is in early compensatory growth. So because kidney is quite a complex organ, and it comprises of millions of nephrons, and each of these nephrons consists of these individual cell types. So we have podocytes and periapital epithelial cells in the glomeruli, where it filters, do the filtration, and all the filtrate will go through the proximal tubules and turn into a urine. So at that point, so we are trying to understand whether the cholesterol signature is only found in just a cell type or multiple cell type in the kidney. So to get a spatial definition, we went on to use this exciting single cell technology to try to dissect this apart. So briefly, with this single cell technology, what we've done is we collected the kidney at 24 hours because that's where we saw all the cholesterol signature is. We then do a sectioning of the kidney to where it includes all the cortex, medulla, and endomedulla. We then isolated out the nucleus, which contained the transcriptional identity of the cell, where they come from, and we then feed it into this 10x genomic instrument that we are lucky to have in Flinders on site. And so what it does is the nucleus go into the instrument, and it forms an oil droplet containing all the reagent necessary to barcode all the genes in the nucleus. So we then perform sequencing in order to digitize this information for downstream data analysis. So here is a plot. Sorry, I'm not sure why all the notation is missing, but here is a plot of showing all of 17 nuclei that were used to generate this plot based on the pattern of the gene expression in each of these single cell. And here in each dot represent a single cell. So here we are able to see some podocytes, parietal epithelial cells, which is all coming from the glomeruli. And we have proximal tubules here, which is this. And the first thing we really find it striking is that we saw a change in the proximal tubules when there's a separation of the proximal tubules between the nephrectomy mice and the sham-operated mice. So when we look at the number of gene expression changes across all the cell types in the kidney, the two major changes were the proximal tubule cells, which are important in filtering and sodium and water reabsorption. And what's striking here is the second most important, most changes were the thick ascending loop from the medulla. So the next thing we're trying to do is trying to find what all those genes means. And so again, so what we can see here is that there's a lot of metabolic changes in the proximal tubules where there's increased glycolysis, and which is shown here, and there is also some response to hypoxic signature due to the increased workload in the remaining kidney. So what's really striking here in the second most change cell type were the thick ascending loop in the medulla. We saw a striking increase in the cholesterol signature here and the mTOR signaling. So if we weren't doing the single cell, we wouldn't be able to tell where this cholesterol signature is actually coming from. So this is really exciting for us. We are sort of following up on that, trying to see which cholesterol is actually in there. So the powerful thing about single cell is that we are able to look at cell-to-cell communication by looking at the single cell data. And here is just here's just an example of the overall signaling changes between the nephrectomy mice and the sham-operated mice. And we can see, because besides cholesterol, we are also interested in IGF signaling, because IGF signaling has been shown to be increased in circulation when after performing nephrectomy. And here, when we see in a normal intact kidney, we can see that the majority of the IGF signaling is coming from the endothelial cells in the glomeruli. And whereas in the sham-operated mice, this is when we actually seeing that the parietal epithelial cells starting showing signaling to other cell types other than the endothelial cells here in the glomeruli. So which is really exciting, and now we are following up on that and trying to look at other signaling pathways as well. This is just an example of what we can do with the single cell data. So with the CRH model that we set up, using this single cell, we have gained quite a lot of knowledge on how it works, and so what we're trying to do next is also what this single cell can be used to do is also to look at other kidney problems, such as the effect of drug treatment in individual kidney tissues. And obviously, there's a broader array of use of this single cell technology to look at other tissues and disease contexts. So with this single cell technology, we have identified 34 kidney clusters based on gene expression, and we saw the greatest change in the proximal tubules and also the thick ascending loop of Henle in the medulla. And we uncover a unique specific proximal tubules that has increased in glycolysis and response to hypoxic condition. And in the mTau, we show an increase in cholesterol signature and mTOR signaling, which we pin down using the single cell technology. So in the medical mind, cholesterol, people think cholesterol is bad. So what if this is important in the kidney hypertrophic growth? So the question we want to ask is this cholesterol really needed for this healthy growth? And what if we could manipulate cholesterol in the animal and really affect hypertrophy? So thank you everyone from the lab for supporting this work. Thank you.
Thank you, Dr. Wong, for yet another great presentation and also for a demonstration of how to think on the spot and react to everything. And next up is Mr. Evan Jenkins, a biomedical engineer in the Cardiac Signal Analysis Lab. The title of Mr. Jenkins' presentation is The Inspection Paradox, an important consideration in the evaluation of rotor lifetimes in cardiac fibrillation. Thank you.
Thank you. So, I'm Evan Jenkins. I'm a PhD candidate with the Cardiac Signals Analysis at Flinders University. So, I'm going to talk about a study we recently performed titled The Inspection Paradox, an important consideration in the evaluation of rotor lifetimes in cardiac fibrillation. So, a bit of background. When I say cardiac fibrillation, I'm referring to two arrhythmias. The first is atrial fibrillation, which is disorganized electroactivation of the atria. So AF is the most common sustained arrhythmia, affecting up to 33 million people worldwide. And the other arrhythmia is ventricular fibrillation, which is very disorganized activity in the ventricle. So VF doesn't affect as many people as AF, but it is the leading cause of sudden cardiac death. And although these are two distinct arrhythmias, they are both characterized by chaotic and seemingly disorganized electroactivity in the atria and ventricles. So a characteristic observation of AF and VF is the presence of these continuously regenerating reentrant circuits, known as rotors or phase singularities, PS. So you can see them in the images on the screen. So on the left is an optically mapped phase map of RAT VF. So you can see this point where the electroactivity is circling around. And on the image on the right, you can see human VF, where again, you see these reentrant circuits forming with the phase progression around it. So our group's recent publications have shown that phase singularity regeneration can be modeled as a renewal process, with further studies extending this to explain that the PS population dynamics in AF and VF can be explained with equations. And this is due to the exponential shapes formed by the distributions of PS lifetimes and interformation times. So this is relevant because for this study, because an inspection paradox is a common issue that can occur with renewal processes. It can arise due to issues in how events are recorded or observed. So it can arise due to an overemphasis on visibly more significant, larger, longer events. So in the case of AF and VF, there is a potential of overrepresenting phase singularities with longer lifetimes relative to the statistical mean lifetime of all PS across a full recording of cardiac of AF or VF. So we hypothesized that an inspection paradox could influence the perception of phase singularity lifetimes in the early stages of cardiac fibrillation recordings, leading to an overemphasis on the importance of long lifetime phase singularities. And we characterized the effect of a potential inspection paradox in 8 systems of human, animal, and computational fibrillation. So the computational simulations were first 10 based on the Aliev-Panfilov cell model, 4 based on 2-dimensional Court-Manche-Tel simulations. A 3-dimensional simulation based on the Court-Manche-Tel model was also used for validation. For atrial fibrillation, 3 cases of optically mapped rat AF were analyzed, 10 cases of human AF mapped using 64 electrode Barster catheters within the atria during EP studies, as well as another 10 cases of human AF mapped using 16 electrode HD grid catheters. For ventricular fibrillation, we used 10 cases of optically mapped rat VF obtained under experimental conditions, and human VF mapped using a 256 electrode epicardial sock wrapped around the ventricle during cardiac surgery. So the lifetimes of all phase singularities across full epochs in each case were examined and recorded, forming the exponential distributions characteristic of phase of exponential renewal processes. And we simulated potential commencement of recordings within these epochs. So we examined the lifetimes of phase singularities active at randomly selected time points within the recordings, and this was repeated 10,000 times to develop a distribution of phase singularity lifetimes representative of what you would see at the initiation of a recording, or if you were to suddenly pick a random point to look at in a recording, such as during real-time analysis of the ventricles or atria during EP studies. And the distribution of phase singularity lifetimes in this sample were compared with the true distribution, which you would see across a full recording of AF. So in the computational simulations, the lifetimes of phase singularities at the simulated commencement time points were on average 84% larger than the lifetimes of the mean lifetime of the true distribution. For the Court-Manche-Tel model in 2 dimensions, the mean phase singularity lifetime increased by about 600, 700%. In rat AF, mapped using an optical system, the mean increase in PS lifetime was about 300%. In human AF mapped using Barster catheters, there was a mean increase in the PS lifetime of about 130%. And in human AF mapped using HD grid catheters, the mean increase in PS lifetime was again about 140%. So very similar for the human AF. For the VF, for the optical map VF using rats, there was a mean increase in the PS lifetimes of 130%. And for human VF, there was a mean increase in the PS lifetimes of 117%. So overall, across all systems, we found that at a potential commencement point for a recording, the phase singularities would be well above the statistical mean phase singularity lifetime that you would see across the standard distribution form for sampling every PS across a full recording. So the implications of this are that at the initiation of recording of AF or VF, phase singularity lifetimes are disproportionately likely to exceed the statistical mean PS lifetime of the full recording or epoch. And this may contribute to a cognitive perception of temporal stability of phase singularities or rotors. And this is important as purely visual inspection of AF or VF would tend to fall on a period of extended re-entry, such as during procedures aiming to ablate re-entrant circuits as potential drivers of fibrillation. And this is an important potential source of bias in phase singularity studies. And to avoid an inspection paradox, careful consideration of the full distribution of phase singularity times across an epoch is required. Thank you.
And thank you, Mr. Jenkins, for this interesting talk. So now we hand over to Mr. Roberts, and Mr. Greg Roberts is a pharmacist with great interest in clinical research who will give a presentation entitled The Glycemic Ratio Towards a Better Understanding of Mild Glycemia in the ICU. Thank you.
Thanks and good afternoon. I'm really pleased to share with you some exciting results from my involvement with a most excellent international collaborative. The purpose of this collaborative is to fully investigate an extensive critical care database collected by Jamie Crinsley, who's a New York-based intensivist. The group shares a common interest in stress-induced hyperglycemia, and this is the sudden increase in blood glucose that's associated with any sort of acute physiology, so typically seen at hospital admissions. It doesn't care whether you've got diabetes or not. It's across all medical diagnoses and across all surgeries to varying degrees. The association with poor hospital outcome is well known. In ICU, it's increased mortality. Their increased infection rates are quite marked, and this is particularly post-operatively. And there's also an increase in thrombus-driven events, so we see poor outcomes in hospital patients with myocardial infarction and ischemic stroke. 6 randomised controlled studies looking at blood glucose intervention have been consistently inconclusive and confounding over a 15-year period. 7 years ago at Flinders Medical Centre, we introduced the world to the stress hyperglycemia ratio. So this is the proportional change from your pre-admission glucose. And this was enabled from this simple HbA1c based equation, which gave us an insight into the pre-admission glycemia. So the stress hyperglycemia is just basically your admission glucose over the estimated glucose before you came in. And to take you through it, you see if a patient with HbA1c of 6 comes in, this implies a pre-admission glucose of 7. If their first blood glucose is 10, then the stress hyperglycemia ratio is 1.44, 44% above their pre-admission. Likewise, a person with HbA1c of 10 implies a pre-admission glucose of 13.3. If they have that same blood glucose of 10, that implies a decrease from pre-admission of 25%. So 2 different, same blood glucose, but 2 very different stress hyperglycemia ratio values. In ICU, we're now aware of 3 different types of malglycemia. So hyperglycemia, as well, and I say that's been scattered around a little bit, obviously. Absolute hypoglycemia, any blood glucose less than 4. And more recently, from a Melbourne group, it's relative hypoglycemia. So this is the more than a 30% decrease in your pre-admission glycemia, and it seems to be restricted to those with very high HbA1c. There remain quite a number of unresolved issues around how it all fits in. For this particular investigation, we renamed the stress hyperglycemia the glycemic ratio, and this is an acknowledgement of the fact that it's now actually bidirectional in nature, the malglycemia. but also because of our way that we were going to utilise it, and that was across the whole admission rather than just a single point at admission, but across the whole length of stay within the ICU. So observational from Jamie's 20-bed ICU. The point of distinction here was the 3-hourly blood glucose levels that they did routinely over this period. So it was provided exceptional granularity. We included these patients, got rid of those, leaving us with just under 5,000 patients. Our mean variables of interest were the mean blood glucose over the entire length of stay, and the glycemic ratio, which was just the mean blood glucose divided by that pre-admission glycemia. Primary endpoints for mortality, and we also looked at risk-adjusted mortality, so adjusted for Apache 4 predicted mortality. Patient metrics were unremarkable. 20% with H1C above 6.5, an in-hospital mortality of 12.6% on a background of an Apache 4 predicted mortality of 20%. Over half of the patients got interventional insulin at some stage, and admission glucose and admission glycemic ratio were about 12% higher than the rest of it for the length of stay. Looking at the association of the glycemic ratio and glucose with mortality, we see two very, very distinct patterns. So here there's a J-shaped curve for glycemic ratio, and over here is a largely linear response. Of interest, there's clearly a sweet spot between glycemic ratio 0.8 to 1 for these patients, and interesting, the highest blood glucose category was associated with a decrease in mortality. Now, the pattern of HbA1c here is really important. For the lowest glycemic ratio with increased mortality, we see that is very high HbA1c with quite normal-looking blood glucose levels, so the domain of relative hypoglycemia. As blood glucose, as the HbA1c increases along here, mortality increases again, and it's very low to normal-looking HbA1c, so mainly non-diabetic patients rather than diabetic patients. Over here, the pattern is the opposite, so for glucose, we start at low HbA1cs and it just increases, and it tells us that a lot of what we're seeing is actually related to the pre-admission glycemia. It's not just pulling in stress hypoglycemia, but it's also the pre-admission glycemia. And we've got both the risk-adjusted mortality there and the absolute mortality, no difference between the two. This is what it looks like if you use the glycemic ratio 0.8 to 1 as the sweet spot. We know from continuous glucose monitoring AUCs that a non-diabetic patient, that a glycemic ratio of 0.8 represents the fasting glucose. So this range of 0.8 to 1 represents probably fasting glucose to about 25 to 30% above that, so just for perspective. Regression analyses, a single value at admission wasn't related at all. No association. When we look at it across the entire length of stay, the glycemic ratio was strongly associated with outcome. Glucose also remained independently associated, We looked at the hyperglycemic aspect separately, so the GR above 0.9, which was our trough, you'd remember. And in that analysis, glycemic ratio was independently associated and glucose not. Neither was associated when we looked at the hypo component of it, although the glycemic ratio was actually strongly independently associated in unadjusted analyses. We used locally weighted scatter plot smoothing to look at subgroups of interest. And you'll see here just a single admission glycemic ratio didn't give us the prognostic acuity that the mean over the length of stay gave us. And really nothing to distinguish those two for blood glucose. We're interested in low and high haemoglobin because the low haemoglobin may represent anemia that falsely decreases your HbA1c value and therefore giving a falsely increased glycemic ratio. You can see here that while the shape of the curves are identical, it did move the curve across by a factor of about 0.1. And again, for HbA1c, there's really not much to see. Finally, and importantly, this slide's really critical. You can see here it demonstrates the ability of the glycemic ratio to very independently pick stress hyperglycemia regardless of your pre-admission glycemia. Whereas blood glucose, it displays quite nicely that blood glucose cannot do that, does not do that. So relies on the pre-admission glycemia. So in conclusion, the glycemic ratio certainly enhanced the sophistication with how we can view blood glucose levels and MEL glycemia in the ICU. The glycemic ratio target of 0.8 to 1 certainly needs another look. There was relative hypoglycemia apparent in those with high HbA1c and low, normal to low blood glucose. There was relative hyperglycemia, so interesting, mainly the domain of non-diabetic patients, and often at BGLs below 10, which is our conventional interventional threshold. The harm from the MEL glycemia was cumulative across the admission, and we've subsequently completed 3 more papers using the glycemic ratio to look independently at hyperglycemia, hypoglycemia, and the temporal relationship across the ICU length of stay. And the data we've found from that is really quite amazing. It will certainly provoke a major rethink in this space, and I think ultimately a change in clinical practice, which hopefully would be associated with improved mortality. But that's in the future. We'll wait and see. So thank you for your attention.
And thank you, Mr. Roberts. And last but not least, we have Ms. Sarah Madigan, a respiratory scientist in the Department of Respiratory and Sleep Medicine, presenting on assessment of cardiopulmonary exercise, testing safety and feasibility in patients with systemic sclerosis.
Thank you for coming today. This is one part of my PhD, but it is an important part, as a test needs to be both safe and feasible in a patient cohort to be usable. So my talk today is going to cover a bit about background, how my patients can participate in the study, the questionnaires I'm using to assess the safety and feasibility, my patient cohort, my results, and my conclusion. So systemic sclerosis, or scleroderma, is a multi-organ disease characterised by inflammation, vasculopathy, and interstitial fibrosis. Now, 5 to 12% of all scleroderma patients will go on to develop PAH, and this is a debilitating disease with poor patient outcomes. And it's actually listed as one of the main causes of mortality in this patient cohort, contributing 30% of all scleroderma deaths to it. So patients are screened yearly for the presence of PAH, and although the current screening methods have a very high sensitivity, there is a poor specificity. So my PhD is looking to assess if the use of a cardiopulmonary exercise test, or a CPET, can screen patients for PAH in a clinical cohort of scleroderma patients. And historically, people have been very hesitant to use a CPET on patients due to a perceived risk of poor patient outcomes, especially if looking for something such as PAH. So my clinical cohort is patients that, as long as they meet the ATS-ERS safety criteria and attend the rheumatology clinic at the RA or here, they're asked to participate in my study. And what happens when they do participate is they come to Flinders and they complete a maximal CPET, and they complete 2 questionnaires on the day of testing and 3 questionnaires post. We can see here, this is my patient. Sorry. The mask allows me to measure any ventilation and gas exchange that has happening during the exercise. Underneath is an ECG to monitor the heart rate and rhythm. The patient has their blood pressure monitored as well to make sure that everything's going as we would expect. And for the patient, it just feels like they're riding up a steeper and steeper hill. They'll get to a point where their legs won't continue or they won't be able to catch their breath. That's our maximal point, and the load's taken off and we watch them recover. Now, mine is a longitudinal study where patients are invited to come back and complete a CPET at their next time of screening, which is approximately every 12 months or when clinically deemed necessary. And my talk today is about how we assess the safety and feasibility of the CPET in this patient cohort. And we did that using questionnaires. So the first questionnaire is the consumer feedback. This is just a sample. This is an in-house developed questionnaire where we're asking patients questions such as how willing they are to come back. In the days following the CPET, was there increased things in such as muscle pain or fatigue? And did they have to seek any medical intervention in the 3 days post-CPET? The other two questionnaires, the EQ5D5L and the Scleroderma Health Assessment Questionnaire, or the SHAQ, are both internationally used health assessment questionnaires that have been validated in the scleroderma population. Both of these questionnaires are regularly used to monitor disease changes in this population, and they allow us to see how things are happening over time. So the EQ5D5L has 5 different domains, and the patient's asked to select on the day what level they feel their health is in each domain. They then go on to complete a visual analogue scale where they're asked to rate their health on the day. 0 being the worst health they can imagine and 100 being the best. So the EQ5D5L and the scleroderma, or the SHAQ, are both filled out on the day of testing and then 3 days post, and the consumer feedback is only filled out after. Now the SHAQ is a different questionnaire. This looks at 8 different domains which make up the Health Assessment Questionnaire Disability Index. We can see here the first 4, and the patients asked over the last week, how do they think their disease has affected them? So for example, when they're dressing and grooming, are they able to shampoo their hair? And they select without, with some difficulty, or unable to complete. They also go on to complete 6 VAS that are scleroderma specific. So it's a very interesting disease because it covers more than just one area. So they're asked to rate over the past 7 days how they think their scleroderma has affected their pain, how it's affected their limitation on an activity, if they've got gastrointestinal problems, their breathing, Raynaud's, and their finger ulcers. So these are all things that commonly occur in scleroderma, and we want to see if it's affected over the past 7 days. And on this scale, 0 is no problem at all, and 100 is as bad as it can be. So overall, at the time of my abstract, 48 patients had enrolled in the study, and the majority of these patients were female because it affects more females than male, and the median age was 58. Now, not everyone completed their surveys and sent them back, so the remainder of my talk focuses on the 39 patients who did, but there's no change in my population. So the willingness to complete another CPET, everyone but one person was either willing or very willing, and that one person was neutral, and they have come back and completed their second CPET since. 2 patients did seek medical attention after the CPET. The first patient had their standard rheumatology appointment with their rheumatologist and mentioned, I've had some muscle pain after. The other patient actually went to their GP for chest tightness and increased in breathlessness. However, the GP didn't follow up any testing as it was deemed just to be a small matter, so no further testing required. Looking at the EQ5D5L results, now this is where the patient rated their health. So 0 being the worst health possible and 100 being the best. There's no change between the CPET before and after, although patients do seem to be rating their health, majority of patients rating their health more towards the healthier end of the scale. Now looking at if they had any difficulty completing their usual activities after the questionnaire, after the CPET, and again there was no change for these patients. Moving now to the SHAQ. Again, this is the disability index. So this is where patients rated the 8 different domains how they felt their illness was affected. 0 being no problem at all, and 3 being the worst health they can imagine. Before the CPET, the median score was 0.125, and this actually dropped to 0.063 after the CPET. So we have a significant change, and this change actually shows that patients felt their disability was less after the CPET than before the CPET, so they felt better in themselves in the 3 days post. When we look at how they rated their severity of pain, 0 again being no problems, 3 being the worst, and we had no increase in pain after the CPAP. We did find a significant increase in breathing. Patients actually felt less breathless after the CPAP. So our medium score before was 0.22 and after 0.40. So they felt better in their breathing after they completed their CPAP. And looking at how they rated their overall disease limitation, again, we see no significant change, although there is a trend to patients feeling that maybe their disease was a bit more of a difficulty after. But most importantly, we had no adverse events on the day of the CPAP, and although I've only shown you a selection of the results that we've got, we didn't see any significant change in any of the other measured parameters, except for digital ulcers, pain, which did increase after the CPAP, but this is thought to be from the patients gripping the handlebar as they ride up. So we can see that patients are willing to complete another CPAP, and we do have patients coming back for their second. We can see that there was a trend to an overall improvement in the EQ5D5L when they rated their overall health. We did see a trend that the disability index actually decreased after the CPAP, so that they had a significant improvement in how they felt in themselves. We showed no increase in pain or overall activity limitation, but we did see a significant improvement in how patients rated their breathlessness, so they felt less breathless after the CPAP than before. So my conclusion is that it seems that the CPAP is a feasible test in this patient cohort, which is safe and well tolerated. We saw no significant decline in any of the health scores that we assessed, but we did see a significant reduction in the disability index and an improvement in the breathlessness after the CPAP. And we do have patients coming back for their second CPAPs now. So I'd like to thank my supervisors, Professor Adams, Professor Proudman and Dr. Davies, the Department of Sleep and Respiratory Services for their ongoing support, the Rheumatology Department, both here and at Carlin for patient recruitment, the Respiratory Function Unit, especially David Shembry for their ongoing support, and all my volunteers who turn up and complete these tests. Thank you.
And thank you, Ms. Madigan, for presenting this great research. This was our last presentation for today, and now I would like to invite all the presenters on stage for the Q&A session. We might start with one of the online questions. So this question is for Dr. Wang. Any downside to kidney hypertrophy? Should we be thinking about kidney days at the gym? This is for Dr. Wang.
Oh, sorry again.
Someone online asks, any downside to kidney hypertrophy? Should we be thinking about kidney day at the gym?
I think because even with one kidney, they can just live like a normal life. So even going to gym shouldn't be a problem, I believe. Yeah, that's my thought at least.
So is there anyone in the room that would like to ask questions to our presenters? If not, I might have a few questions. One is for Dr. Chan. So what would be the best steps for the translation of your findings into clinical practice?
Yeah, so we're trying to look into the collagen mineralization that could be a potential biomarker for identification of patients. Okay. In that sense, yeah.
So, yeah.
More towards therapeutics, yeah.
And yeah, another one.
So well done all on some very diverse presentations, and you all did well to battle with some of the technical challenges. I was going to try and answer a question to capture you all, but it's very difficult. But the question I'm going to ask is around the paradox, the inspection paradox. So this was new to me, and I just wonder if you could help me understand does it cause trouble in other data? So we've seen some other time-related data sets here. Might we be neglecting or not completely capturing data by virtue of the inspection paradox that you talked about in relation to rotors? Can you just help me understand, does it have a broader issue in data interpretation?
Yes, so the inspection paradox, in the context I was discussing, it's a common issue with renewal processes. So typically this would be more sort of standard statistics and sampling. So if you're trying to figure out the average class size at a university, there are ways, if you go to, so it typically only arises when you are sampling data in a way that introduces a bias towards largest. I'm trying to remember the examples that I've. So if you're sampling class sizes at a university, and you were sampling students as they left, around, let's say, 3, you're more likely to run into students who have just left a very large lecture class than students who are in a small workshop. So you'd hear more people saying their class sizes are huge rather than what the actual number would be if you were to speak with the dean of the college. So typically, in the context of cardiac fibrillation, which I was discussing, it's mostly an issue during very short recordings or the initiation of it, especially when you are, so at the start, you're very likely to see a very active phase singularity, probably one that's very large as well if it's staying around for a longer period of time than the statistical average lifetime. Most of the time, it's not going to be an issue in most time series sort of data, but just you have to be aware that you're not introducing some form of unintentional inadvertent bias towards longer or more significant events. Yep.
And I have another question for Ms. Roberts. You were briefly mentioning to this at the end of your presentation, but have you looked at other endpoints like length of stay or infections rate, other than mortality?
Jamie's database at this point only has in-hospital mortality. We have the third of the subsequent papers we've done has looked at the temporal relationship across the ICU length of stay, and that's been quite revealing. So it looks like we introduce relative hypoglycemia in those with high HbA1cs, and this probably causes a lot of harm, and it appears this is through our conventional management. So the patient with HbA1c of 10, they're running a background blood glucose of probably around 15, and the first thing we do when we pull them into ICU, of course, is give them intravenous insulin and pull them down to about 8 or 10. So they're literally guaranteed to have an excursion below a GR of 0.7, which is the induction of this relative hypoglycemia. So going back to the second paper that we still haven't published, the hypoglycemia, the relative hypoglycemia is actually related incredibly to excursions below 0.7. So it's not the time below, which is everyone was fishing around, but it seems to be maximised when you've got at least a 24-hour non-exposure period where you're not below 0.7 and something recharges. So the next time you have a hypoglycemic episode, it's a lot more potent. And as you increase that interval of rest between the hypoglycemic exposures, the odds ratio for mortality just goes up and up and up. So it looks like there's this recharging process for the counter-regulatory response when you have a hypo. And we're not sure why it only happens. We only see it in HbA1c above 8. There's not even a sniff of it before that. That's absolutely consistent with the Melbourne group who published some stuff about a year ago. They only saw it in HbA1c 7.5. So we don't have any stuff on infections, but some stuff I'm just writing up now from here looked at the association of any exposure with hyperglycemia defined as a glycemic ratio above 1.1. That's the first of the new papers we haven't published. There's a threshold at 1.1, and above that, you start getting very significant negative outcomes. So any exposure above 1.1 was significantly associated with hospital-acquired pneumonia from here. And that was adjusted for anything you want to look at, pre-existing respiratory disease and length of stay, age, all of the classic sort of variables associated with hospital-acquired pneumonia. So this whole, looking at the whole thing through this prism of glycemic ratio, all of this stuff has just become sort of wonderfully clear. We've just got to yet publish it, and I won't get too far ahead of myself, but I think there'll be a lot of changes over the next few years in that space.
That's fascinating. And I also have a question for Ms. Madigan, which actually are two questions combined. One was did you have a lot of difference in between the length between the first and the second assessment, or was that pretty consistent for your participants? And also, I would like to know what were the biggest challenges for patient recruitment, and how did you overcome that, if there were any?
Every patient has 3 days. So my patient today gets their first sample questionnaire today, and they fill them out on Sunday and then post them back to me.
I meant in between the first appointment and the second appointment, when you did the repeated.
When they're coming back, I'm just starting to get the second CPETs through now. So patient recruitment, I only started in April last year. So there's a bit of a lag sometimes between when they see their rheumatologist and I can get them in, but we're averaging between 10 to 14 months between the two CPETs. And we'll start presenting that data later next year, looking to see if their disease has increased, has the impact of the CPET changed. So we don't have a standardised number. We try to tie it in the second time of screening, but patient recruitment occurs the first time they see their rheumatologist. My biggest problem with recruitment is that the scleroderma is centrally managed at the RA, and then I'm having to tell patients, well, I'm actually based at Flinders. It's not a hospital that they know very well. Parking isn't the easiest. I don't think it's easy at the RA either. So encouraging patients to drive another 30 minutes, sometimes past their normal hospital, and explain where all the parking is. We're also having some problems recruiting country patients because they see their rheumatologist on a Monday. Because I work part-time, I offer the CPET on a Thursday, and they don't want to hang around another couple of days. So the department's really good, and we're being more flexible at trying to fit country patients in so that we're getting a true representation of our cohort. But my supervisor is the head of rheumatology, and she's a very convincing recruiter. So I'm doing really well. I've got 54 patients tested now and another 40 coming through, so I should hit the 100 mark in the not too distant future.
Sharing this pain, I actually find it really impressive that in 1 year you got so many patients in, so congratulations. Is there any other question from the floor? So I don't believe I've seen any more questions online. Let me double check. Doesn't look like, no. So if not, I would like to thank you all for being here. Oh, sorry. Sorry? Is there someone with a question? Sorry.
Don't worry, that's fine.
Then if there's no more questions, again, I would like to thank you all for being here. Thank you to the presenters. Thank you to the audience. And I would like to remind you to express your vote in the polls on VUA. And there's some refreshment and afternoon tea in Lecture Theatre 3. Thank you. Bye.
Okay, good afternoon everybody, and thank you all for coming. I know it's a bit late in the day, so we appreciate your attendance. We've got some exciting speakers about to present, which is very exciting. But firstly, I just want to quickly say that we acknowledge that the Kaurna people are the traditional custodians of the Adelaide Plains, and that we pay our respects to our elders past, present and future. We also recognise and respect their cultural heritage, beliefs and relationship with the land. And we also acknowledge that they are of continuing importance to the Kaurna people living today. So firstly, I'd like to present Associate Professor Andrew Vauquelin. And I'll just, while you make your way to the front, I just want to give a quick background. So Professor Vauquelin is an NHMRC Career Development Fellow within the Adelaide Institute for Sleep Health here in our College of Medicine and Public Health at Flinders University. He has a research interest and expertise investigating the impact of sleep loss and sleep disorders on daytime sleepiness, sleep neurobiology and neurobehavioural function, particularly in relation to operational performance and motor vehicle accident risk. He has also been leading the development of innovative online sleep health decision support tools and digital sleep health solutions, utilising new sleep monitoring technologies for simplified screening, diagnosis and management of sleep disorders. He increasingly works with industry partners on translational sleep health projects and chairs the Australian Sleep and Alertness Consortium Steering Committee and the Sleep Health Foundation Consumer Reference Council. So very happy to have Andrew here today. Welcome.
You could probably go up here. Thank you. There's no slides, is there? Oop. Sorry. No worries. No, that's fine. Mine didn't make it. That's all right. Thanks, Maddie. I'll talk through it. That's all right. Thanks, Maddie. Yeah, and look, I also just want to acknowledge that we meet on the lands of the Kaurna people and acknowledge leaders past, present and emerging. So, look, I guess I just wanted to do a very quick introduction to who I am and sort of what I do and my experience so far in sort of working a little bit with industry and translational sort of research and how I'm finding it at the moment. So I'm a mid-career researcher now, so I've done my degree at Adelaide University, did a postdoctoral work at the Wilcock Institute in Sydney. And yeah, sort of Maddie alluded to, my work's been sort of around looking at daytime outcomes in sleep disorders, so sleep deprivation and sleep disorders and how they impact sleepiness, particularly in operational environments. But more recently, I guess my experience started to involve industry a lot more, and really not myself going out finding industry, but my experience, at the Wilcock Institute when I was a postdoc, they were part of a team that have a successful cooperative research centre. So it was a CRC in alertness, safety and productivity. So me being a naive sort of younger postdoc, not have anything to do with industry, just on a traditional pathway, suddenly got exposed to a very large, or for me it was a large consortium of industry partners, multiple academic partners. I was fortunate to collaborate and work with Karen Reynolds, College of Science and Engineering, who was part of the CRC. And that was, it was a completely different way of looking at things, working with industry, understanding what they actually want and how they view the world, which is really from a completely different perspective. And so for me, I've quickly sort of learned that they look at it from a different perspective. They want outcomes that are obviously driven by business or driven by outcomes that are obviously impacting business, where we as academics look at things like publishing, doing quality research. Our timelines are much longer. Our expectations are quite different. But either way, I found it quite rewarding learning about that. And so my research at the moment is all around three main themes. One theme is looking at
predictors of operational performance and alertness failure. So that's basically, can we identify people either with sleep disorders or sleep issues who are at high risk of crashing a car or having issues at work? At the moment, that's very difficult. It seems straightforward, but it's actually very hard. The other theme is really playing a lot with technology. As sleep, at least in the last 5 years, it really lends itself quite well to new tech. Everyone's got their Fitbits, everyone's using techs and their phone, where in the past measuring sleep is quite complex, you need a lab, wires everywhere. Now everyone can sort of measure sleep with their technology. And so, at first it was resistance, and we sort of like, this is old gimmicky stuff where, we're real scientists and clinicians, we don't believe this stuff. But now there's a lot more buy-in, and the technology is reaching a point where it's quite mature, and that again opens doors working with industry. So I work a lot with partners that produce and make and try to validate and develop technology that transforms our bedroom into sort of more smart environment that can measure sleep over a prolonged period of time rather than a single snapshot. And so from that experience, I found that it's very rewarding. There's been some success in commercialization, there's some success learning about IP, and that sort of pathway. And so I felt it's sort of enriched my career, and certainly, I enjoyed working in that space. I might stop there. Yeah.
Thanks, Andrew. Sorry about the slide. I'm not sure where that went. Okay, so we'll leave the questions for after all the speakers have presented. So I'll introduce our next speaker, Professor Karen Reynolds, who's from the College of Science and Engineering. And Professor Karen Reynolds is director of the Medical Device Research Institute at Flinders University. She's passionate about bridging the divide between research and industry. And in 2008, she founded the Medical Device Partnering Program to facilitate early-stage innovation and collaborations across the medtech sector. Karen is a member of the Therapeutic Goods Administration Advisory Committee on Medical Devices and on the board of the National Imaging Facility. She's a fellow of the Australian Academies of Technology and Engineering and of Health and Medical Sciences, and is an honorary fellow of Engineers Australia. Welcome, Karen.
Thank you very much. So I've actually got a slide, I think. So there's a bit of a difference between myself and Andrew, is that I've been around a lot longer than Andrew, and I've got a time frame here which lasts about 35 years. So it's going to take a quick snapshot of what I've been doing to really talk about my career pathway. So I guess the first part of my career was in the UK, where I was brought up, and I spent a few years at Oxford, both in the university and in the John Radcliffe Hospital, and started doing research, I guess, having done a physics degree but wanting to get into biomedical engineering. So much of my research then was around physiological monitoring and sensing, so optical sensors primarily. From there, I moved up to Leicester, and I had, again, it wasn't postdoc, I hadn't done my PhD at that stage. I did my PhD in Leicester University, looking at prosthetic heart valves, trying to determine when they might fail. And then I got a job as well in the Leicester Royal Infirmary, and that was, again, looking at sort of sensing and physiological monitoring. So that was really the start of my journey. At that point, in 1997, I came to Flinders, so I've been here for 25 years. And so I guess, again, the first sort of 10 years or so, I'd say a relatively traditional kind of pathway. Again, just doing research, getting some success in some grants. My first ARC grant there was looking at, again, optical sensing. So it was a bilirubin monitor for trying to detect jaundice, really, in newborn babies. And then started to move a little bit. I got interested in surgical instrumentation. So again, instrumentation and monitoring, but could we actually sort of help develop kind of smart instrumentation? And to do that, it needed to measure things about. So we were trying to put screws in to bones using a smart screwdriver, for example. So that then led me to try and understand a little bit more about how implants interact with bone. So then we got into the kind of imaging side of things. So you can see my interest is starting to spread very wide. I also had a program in medical simulation. So we had a company that we set up that was making physical simulators for training in various different medical and surgical procedures. That kind of then expanded into the virtual reality space, of which I had really no knowledge whatsoever, but I had a very talented research assistant who was absolutely fabulous in this space, and we got some significant research funding to support him through that process. So that was a really interesting time. I guess then I started to get then I got a little bit frustrated with the challenges of trying to work with industry, and at that point I launched the Medical Device Partnering Programme. 2008, it's still running. It's the longest running medtech incubator in the country. And we've seen over 700, 800 different projects that have been brought to us by different industry clients primarily, or some of the clinical community as well. And we've developed over 100, or we've done over 120 kind of industry-focused projects. So again, that's been a really interesting journey, really satisfying when you see some of them actually now they're out in the market and being sold around the world. So at this point, my trajectory started getting a little less traditional, I guess. Again, from the research perspective, I got involved in some more bigger kind of research programs. So again, the alertness CRC that Andrew's already talked about. So my role there was sort of the theme leader for the the sort of technology side of things. More recently, there's the first one there is a joint research centre with the China, with people in China, and it was around point of care for again, sort of physiological monitoring and point of care devices held on a smartphone. Part of the ITTC, so Industrial Transformation Training Centre in Medical Implant Technology. I'm part of the new Aged Care Centre that people probably have heard about, a $34 million centre that's now been established at Flinders. Again, my role there is sort of in the partnering, so very similar to MDPP, the Medical Device Partnering Programme, is to try and develop those partnerships with industry. And then we have a global linkages grant very recently as well with Depew and FuseTech, which actually is back in that simulation type of space. So my interests have moved around a bit, but there's sort of, I guess, some themes emerging in there. I guess on a more sort of personal and professional level, I managed to sort of fit in a couple of children on the way who are keeping me entertained. FMDAT was really the precursor to the Medical Device Research Institute, so trying to sort of bring some of this activity together across the university, and that's sort of built, I guess, over a period of years. I gradually kind of got promoted through to my professorial appointment, and then more recently I did things like I was the Dean of Research for the college, so sort of trying to get involved in broader parts of the uni. And then just my last part of this is really then the outside world and taking some fairly significant roles sort of at a national level. So we've already talked about the being a member of the TGA's Advisory Committee on Medical Devices, so providing insight as to whether these new devices coming onto the market are safe. And then I was chair of the Biomedical College for the Engineers Australia, and then some roles within the Academy of Technology and Engineering. 5 years on the Australian Medical Research Advisory Board, which was absolutely fabulous, really trying to help steer the MRFF and where it was going right from the very sort of start of that programme, that funding system. So that's been really interesting. And then just very recently onto the National Imaging Facility Board. So that's kind of my 35 years in a nutshell, and it's kind of a mess, and this is how my life feels most of the time. You know, everything going on all at once in all different directions. But anyway, that's me.
Thanks, Karen. That was very inspiring. Okay, so next up we have Associate Professor Phil Denning, a senior scientist in gastroenterology and hepatology, from Salin and also the College of Medicine and Public Health here at Flinders. Professor Denning currently holds a senior medical scientist position in the Department of Surgery and Gastroenterology at FMC. Prior to this, he held the position of Senior Research Fellow in the Department of Human Physiology at Flinders University and in the School of Medicine and the University of New South Wales. He has research interest in gut disorders and the mechanisms that control gastrointestinal function, and collectively he has published a lot of original peer-reviewed papers, over 150, which is also very inspiring to us. And we welcome you for your presentation. Thank you.
Although after looking at that, Karen, I wonder what the bloody hell I've been doing with my time. I'm going to take this from a slightly different approach, just looking at, I deal with clinical disorders, mainly functional gut disorders, so chronic pain, but also just constipation, incontinence, and looking at how we've built up a picture of what's going on. And with human research, all clinical research, you've got all these stages you've got to go through. Now, we can't do all of them, but if you don't have an idea about any of the other bits, apart from the bit you do, it's really hard to build a picture. I've sort of worked on this principle that part of the challenge of clinical research is to rate the events at the cellular level where they originate to the organ's function, where they sort of matter. So build this picture, and just to give you an idea, this is kind of the direction I've gone in over the last 25 years or whatever it's been. So gastrointestinal disorders. Someone comes to see, they've got a gut pain. We had a technique at St George Hospital where I worked in Sydney where we could record how the gut contracted. But it was a very simple device at the time, few sensors, a long way between. I started working with a fibre optic engineer, John Arkwright, who developed this amazing catheter which completely transformed what we do. We were able to put all these sensors into a gut, we could get detail that we'd never ever been able to see before. The trouble is, this all goes on inside you. You can't see what's going on, so we had to interpret what we were getting. Then I started talking to Simon Brooks, who works at Flinders University up on level 6 here, and they had these beautiful setups in organ baths where we could use our catheters in bits of animal gut. And so that took us to the next step. I moved here in 2011. Suddenly we could do a whole range of studies where I could actually see the gut moving, and we could relate it to the motor patterns we recorded, same catheter as we used in patients, and you got a better idea of the mechanics. That was an animal colon. Nick Spencer upstairs was doing work with the entire human colon sitting in an organ bath. So we were able to do these unique studies where we could record in the patient, they'd then have the operation, have their colon removed, and we could record from the gut in the organ bath and start having a look at how they differed, what role the central nervous system played. So you're pulling together all the pieces to build a better idea. Then you've got this work that's done also in Simon Brooks' lab by some of the people sitting here. This is getting right down to the staining of the nerves. So you can see how a gut moves. You might see the disorders in it, but what controls it? And that's where this stuff comes in. And being able to work on all the individual different components, when you get right down to that level there of staining nerves, working out where nerves come in, what they do, you can start to relate it back to the work you actually do in patients because you're being involved in it. Now, Flinders is just brilliant for this because we have this set up here. I wouldn't have been able to do this at St George Hospital. And then tying it all together, you need software. You need ways of analysing it. And I've employed software engineers that have just helped us to pull this world together so you can see how it all works. But the one problem with Flinders is it's very small in terms of population and patients we see here. So my anorectal clinic might see 100 patients in a year. If you want to build up a big database to look at it, then you need to work with other people. And this is where international collaboration comes in. This is taken from my thing that's on the Flinders page. You have to be willing to give your data to other groups, but the bonus is they give data back to you. We see 100 patients a year here. I work with groups in London that see 1,000 patients a year and they send the data to us. Now we control these data through spreadsheets, databases that are held at Flinders University. But what it's also meant is during the whole COVID phase, where we stopped a lot of work here, this work kept coming in. And I also have the bonus of working in the anorectal clinic, which means I don't need funding for that. I can keep research going without the funding. So where other arms may fall down, you can keep work coming if you've got something that doesn't require the funding. So that's just a snapshot. It's that ability to work with different groups so you build an overall picture of what's going on rather than just narrowing yourself down to one tiny little world. So that's me.
Thank you, Professor Denny. So next up, I'd like to introduce Associate Professor Kate Laver, who's an ARC Discovery Early Career Research Fellow and an occupational therapist at the Department of Rehabilitation, Aged and Extended Care, also within our college. Associate Professor Kate Laver works in healthy ageing here at FIMRI. She co-designs, develops, tests and implements rehabilitation interventions and new models of care which aim to optimise independence and quality of life in older people. She has a particular interest in the use of technology in health services, for example, virtual care, telehealth and virtual reality. She also has a postgraduate qualification in implementation science with expertise in leading large-scaled applied translational research projects. So thank you for coming. We welcome you.
Thank you. There's probably a bit of double up between what you've just introduced me and my slide, but I've got a little timeline here. So I graduated from UniSA in 2002 as an OT and then came straight here as a new graduate therapist. Which was fantastic. I got to see, I know all of these hallways very well because I've taken all the shortcuts and I've seen, thousands and thousands of the Southern Adelaide population. So that was fantastic. And then I think it was around 2005 when the National Stroke Foundation released the first clinical guidelines for stroke. And that was really a turning point for me because, I really felt like having these guidelines which reviewed all the evidence and gave me clinically evidence-based, recommendations on exactly what I should be doing with my patients. It just kind of really put that research practice connection for me, and I started going to more conferences. I enrolled in the Masters of Clinical Rehab here and finished that in 2008. And then, had never didn't like research at university, didn't think about doing an honours, never occurred to me to do a PhD, but I had someone who said to me, look, we're interested in a particular topic, would you be interested in doing a PhD? Didn't even really know it's a career option until I started in doing kind of postgraduate studies. So I finished that in 2012, and then since that as well, I've done some additional work with a graduate certificate through the University of California, San Francisco. So you can see here, I actually did quite a lot of different roles clinically. So I worked here, I worked in slow stream rehab at Julia Farr, I worked at the REPAD, I was in the community, I spent a lot of time doing home-based rehabilitation, which was fantastic, and I did some private practice. So now in my current role, I'm in a research-only position, but I do get to do some teaching, which I like. So I do some guest lectures. I have quite a few research higher degree students who I supervise, which I love, and I do workshops with OTs, which again is really good and keeps kind of that connection to clinical issues. And then, through my position, I do get to do some consultancy teaching as well. I am still doing lots of ongoing development myself. I'm attending conferences, I'm doing online training. The more you learn, the more you realise you don't know. I'm always doing more stuff. I review a lot of journal papers and I do a lot of consumer engagement work as well. And apologies, but I have got my metrics down on the left. I've been writing fellowship applications and it's all about, how many publications you have, how much funding you have. So you can see here, after graduating in 2012, my first fellowship with NHMRC was 2016. So it did take me a few years really to get a lot of work, really build up publications and get a lot of experience before I was competitive for fellowship. And I think my one advice to people to stay in research, as a research-only position is to really be agile. And, I've really gone from my PhD being in stroke rehabilitation, my first fellowship was in dementia, now I'm looking at home design and ageing through my DECRA. So you really have to be a bit agile and respond to community needs and funding initiatives. So some of my recent funding is through MRFF schemes for frailty, for urgent care, and responding to what funders are looking for. And so just a quick summary of what I've been doing. So, I started by loving these stroke guidelines, and since then I've led Cochrane reviews and developed the first clinical practice guidelines for dementia, which were endorsed by NHMRC. I've run clinical trials and, similarly to Andrew, quite interested in the use of technology with ageing and also interventions for people with frailty. And as I said, following on from those studies, have done some translational work as well. So particularly interested in translational work for allied health professionals and looking at things like quality improvement collaboratives so that you can scale up across Australia. And this is, to burst my own bubble really is just to remind people of, it sounds really good when we stand up here and say, I can do this research and I'm doing this and I'm doing that, but it's really competitive. And for all those funding things that, I say I've achieved, there's like 4 times as many that I haven't got. But, we tend to hear about people's successes and not the things. And so the most recent round of fellowship applications is probably around 15%. So you're competing against really good people and, still just have to have a really thick skin. Yeah.
Thank you, Kate. So now we have our last presenter for this session, Associate Professor Belinda Lang, who is a Fellow of the Australian Institute of Digital Health and also the Research Theme Lead of Technology at the Caring Futures Institute, also here at Flinders University. Associate Professor Belinda Lang is a digital health researcher with 20 years of experience in co-design and evaluation of digital health technologies such as virtual reality, virtual humans, and game-based rehabilitation tools in clinical and community settings. She completed her PhD in 2006 and received an American Association of University Women Postdoctoral Fellow Scholarship to undertake research at the University of Southern California in Los Angeles. An experienced physiotherapist, educator, and digital health researcher, Belinda is passionate about partnering with clinicians, consumers, and industry to build digital health capacity and explore how interactive technologies can enhance clinical practice, training, and wellness. She's also the chair of the Flinders University Human Research Ethics Committee and deputy chair of the Southern Ethics Committee. She also holds non-executive board director roles in the Australasian Institute of Digital Health and the Australian Physiotherapy Council. So welcome.
That was longer than it looked on paper. Thank you. Thank you. It's really such a pleasure to be on a panel with such amazing researchers, so thank you for inviting me. Well, as was mentioned, I finished my PhD in 2006. My research was focused on using virtual reality as a distraction tool for children in the emergency department who are having minor medical procedures done. At that time, the virtual reality system that I used was the whole system was around $20,000, and I carried it around in this huge big suitcase, a big travel case. Once I finished my PhD, I got the scholarship to go and do my postdoc. It was a 2-year postdoc at the University of Southern California in the Institute for Creative Technologies. That was an amazing institute. It was a collaboration between the military, academia, and the entertainment industry. So there was lots of opportunities there. And in fact, I stayed well beyond my 2-year postdoc, working there until 2013. And I had the opportunity to work on a whole range of different projects that essentially all focused on co-design and evaluation of different types of interactive technologies, so virtual reality, virtual humans, apps and video games, for a wide range of purposes around well-being, mental health, rehabilitation and training. I also had the opportunity while I was there to travel and go to a lot of conferences, and especially given the current climate, I feel like very, very thankful for that opportunity. Attending conferences is great to get your work out there, to hear about what other people are doing and generate new ideas, and networking as well as the sightseeing. Once I finished at the University of Southern California, I moved with my family to Regensburg in Germany, and I actually took a couple of years just to focus on family. So stepped right away, new place, and really focus on my family until moving back to Adelaide, coming full circle in 2016 to take on my current position. Now, I have a teaching and research position at the moment, and I teach into the physiotherapy program, as well as the health sciences and the honours program. I do have roles within the Medical Device Research Institute and the Caring Futures Institute, and as was mentioned, I work on both the Flinders University and Southern Adelaide Clinical Ethics Committees, and I believe that these roles are really important service roles that give back to the community, but also I get so much value as well in return. I learn so much from my fellow committee members, and the researchers who work that we review, but I do believe that this is just one of the most rewarding parts of my role. In terms of my research, I still work looking at different types of technologies and how we might apply them in practice. So I still collaborate with clinicians and consumers to explore what technologies people are currently using and how we might best leverage the benefits of these technologies to have an impact on clinical practice. My work sort of spans across disciplines, it spans across different client groups, different settings, and different conditions. So I just wanted to show you a few examples. I don't only work in virtual reality, but just to sort of show you a little bit around sort of how far I've come, but yet still working on solving similar problems. But the virtual reality systems that we're using right now cost around $600, and I can carry them around in my handbag. So over the period of time since my PhD, the technology has really caught up with the vision, and we're starting to see that it has become more widely acceptable. So just a couple of examples, we're still using virtual reality for distraction and relaxation across a range of different populations and settings. We're looking at art-based therapy, again, in community and inpatient settings. And the middle one there is a functional assessment and training tool that I'm working with an industry partner to develop with allied health professionals. It's a virtual kitchen, and we're looking at assessment and training that a range of different allied health professionals can use with their clients so they can use that technology together, but for slightly different purposes. So apart from the research, what I enjoy about the work that I do is actually getting out there and working with clinicians and consumers and having that impact on practice, but also working with my HDR candidates and other students and helping to navigate them through the early stages of their research journey. I think that's really rewarding and exciting part of the work that I do. And I just wanted to give a couple of takeaways from, putting the slides together and thinking about the work and my career pathway. I would say just think outside the box. Like, don't let people tell you that, we've always done it this way. Why would we do it another way? When I was using virtual reality, a lot of clinicians and a lot of researchers just didn't see the value in it. And now, they're starting to actually see the potential for this type of technology. And also, again, not underestimating some of those smaller activities that we do. And I do a lot of sort of qualitative, smaller exploratory projects. But these types of projects are the building blocks that actually kind of grow your career and guide your career. It's not always about the really large clinical trials and the groundbreaking discoveries, which are great, but that you can still make an impact with other activities. So thank you.
Yeah, sure. Okay, so I just wanted to say a quick thank you to all our speakers. That was absolutely incredible, especially for early career researcher myself. It was very inspiring to hear everyone's stories. Now we will open up the Q&A. Simon, did you want to hand some? Yeah. So I think we will do the questions in the live audience first, and then we'll go online in a minute. So did we have any questions?
I've got a question, I guess, for Belinda. On the metaverse, it's something we hear about. I remember when I was a kid, Lawnmower Man, and there was I remember there was a virtual reality place on Hindley Street. So, we're at the 6th or 7th or whatever iteration of virtual reality and that sort of thing. But how long until it's, you know, kind of embedded in healthcare delivery and that sort of thing, do you think?
Yeah, I think that's a really great question. We're actually seeing a lot of this type of technology already being used successfully in training of our clinicians and in various sort of aspects of practice. I think the technology is improving so rapidly that it's sort of difficult to keep up with the technology and keep the research up with the technology. So we do want to make sure that all of the things that we're implementing into practice have evidence behind them. And so I think there's lots of opportunity now with the type of technology that we have that it could be used, but we need to make sure that we're applying it appropriately. The biggest thing I think with this sort of technology is not necessarily the hardware itself, but it's the content that we're actually providing through this tool. The technology is the tool, the content is the most important part. So in terms of things like the metaverse and other virtual reality tools and the newer headsets and eye tracking, all of those types of hardware technologies, we need to think about what purpose they're going to serve and how we're going to provide that to serve the people that we are trying to support.
So thanks very much, everybody. And I'm not sure who I'm directing it to, but I just... I was at a conference recently. It was great to get back in and do that, because I think you meet people and have those conversations. They're quite important. And, they were a very famous respiratory physician had died recently, and people were talking about him, and they were doing all those presentations. And one of the guys got up there and said... that Richard Light had said to him, it's better to be lucky than smart, and you're just going to have to work hard. And I guess, and I said, it's sort of okay. But I think I'm sort of interested in reflecting on that, and then what are the system enablers that have been beneficial, because, part of this is not only for people here, but it's also maybe for people in my sort of role, what are the system enablers that stop that conversation from occurring? And really, because, we all know that you've got to be persistent, you do have to be lucky, but we're trying to create a system that actually helps us move it forward. So I'm sort of interested in knowing what they look like and what people, because when people ask me, who's, they want to do a PhD, I tell them, go and find a good, don't worry about it, but go and find a good supervisor. That's the only thing that you have to do, is spend enough time finding a great supervisor, because a great supervisor will have an enabling environment for you. But help us understand what that looks like.
I'll build on your comment around finding a good supervisor. I think regardless of if you're doing your PhD or if you're just taking on other research activities, perhaps putting a toe in the water around doing research, it's really important to have other people around you who understand and you can, throw ideas around with. So that kind of local support structure, as well as the infrastructure that goes with that, the opportunity to have take the time to actually be thinking about what you're currently doing, where you might be going in the future, and then how you're going to get there. And mentors are always a great support, but also when you're thinking about mentors, it's people that can provide some guidance to you, perhaps, but also people that are going to be on your side and put you forward for opportunities. I think that's a little bit around the luck, isn't it? It's building that culture where you have that local support and then the support from others to actually push you forward into other areas.
I'll just add one more thing. I think attending conferences, national conferences, international conferences, and as a clinician, I think just connecting to being part of research culture, connecting to other people, seeing what's done internationally, seeing how it applies, and I think probably more exposure to international conferences. So where the national conference, for example, on stroke, Salin should be saying which allied health people are going, how are we going to be represented at this conference so we can bring those ideas back.
Yeah, I would agree with all that, and particularly I think that whole kind of conference or any other kind of networking opportunity, just get yourself out there, get yourself known. The number of times that I've had maybe students or people who've either I've been approached to say, I've met one of your students at a conference and really good, and what are they doing when they finish their PhD, and so on and so forth. So the networks are so important. So that would be one piece of advice. I guess the other one would be just develop a very thick skin. We get knocked back about everything all the time, and you just take it on the chin and move on, and just also start to think about other opportunities. Again, the number of people who are just wedded to the idea they have to get AS or NHMRC grants. There are other things out there, and if it's just a small bit of money, if you can get things happening, it A, adds to the CV, but B, it does then lead to other opportunities. So,
I guess just one other thing to add: listen to areas outside your field, or indeed don't just go to things in your field, because Karen's whole world developing stuff, there's stuff there you don't even know exists, so you don't even know you've got the problem or there's something to solve a problem till you see something else and go, hey, wow, I could use that. And so I think we do run in the risk of just a focus down our own little narrow path, but when you look at what other people are doing, it just opens the door. And once that door's open, that leads to things and that leads to things. So yeah, you narrow yourself too much if you just focus on the one thing. You've got to get out there and see other ideas.
Yeah, I agree with all of those points. I guess, from my perspective, my experience, I think having really good, strong mentorship and networks around you is sort of really, really critical. And you need to have thick skin, and you need to, keep trying and keep doing all the networking things. But having somebody, at least I found that incredibly, if I didn't have that, I always hear horror stories, and try not to let that sort of put you off completely. Just try and find, if you don't have it in your immediate environment, find a good mentor somewhere else, through a mentorship program. But I think that's really, really important.
We'll do that question first, yeah.
I wonder if you could share the best research career advice that some wise person gave you on your journey. Like for me, it was, Robin, if a PhD was easy, everyone would have one, and that it's just hard work, but you will be rewarded at the end, trust me. So I thought, okay, and so off I went. So what are the pearls that you could share for research career advice?
I'm just thinking of one very brief comment that I got, but sort of stuck with me, I do use it quite a lot when I'm thinking about what to do next, or what to apply for, and other areas of my life. And it was just go for it. What have you got to lose? The grant applications, there's not a very high success rate, but if you feel that you have the right project, you have the right team, you have the right application, and you can give it a good go, then go for it. If you don't, then step back and wait for the next one.
I think, so Maria always says, after grants come out, win some, lose a lot, and so I always think about that. But I think for me it would be being agile and looking forward. So not just what are we doing now, but, what are things going to look like in the future? Because you always have your grants, your research has to be innovative, and it has to be relevant in 3 years, 5 years, because that's how long it takes to get it done. So always looking up.
Yes, good question. I think actually I'm thinking way back to my PhD, and my supervisor, I remember at the time, he said, "Okay, you've got enough, write it up." And I go, "Oh, well, I haven't finished yet. I haven't finished." No, you've got enough, write it up. So I think always realising that you don't have to continue to follow things to the end of the earth, you know, that maybe you've got enough that you can then start to just move slightly in a different direction or chase the next grant or so on and so forth.
Shit happens. Honestly, and when you're dealing with patients, as I think we've all done, you can have the best designed study, but a patient will find a way to screw up that study or you'll have 5 in a row that do exactly the same thing and the 6th won't do it. And you've just got to have that ability to, instead of despairing, well, look what's there, look what we do have. Because any information from patients is so precious that there's always a story there. It's just not the story you perhaps wanted it to begin with. So you just keep going with it and you keep going with it and you keep going with it. Sometimes the door will shut completely, but otherwise, you know, don't be afraid to change the direction if it's not going the way you thought.
Yeah, I think, you know, agility and flexibility, you know, being somewhat opportunistic but having a clear vision is important. So you want to, you know, have a high level vision for your 5-10 years. How you get there might change along the way. And I guess learn to say no as well. I think it's okay to say no. You can't do everything. You don't want to get burnt out. So having a good culture and well-being is really important as well.
There was one question online very similar to the previous question, but slightly different in the fact that what would be your best advice also for someone who's not already in research but about to approach research with these challenges with funding and so forth? So for a new researcher, what would your advice be?
Well, you've got to find a group that's, I think, successful to begin with, preferably if they've already got some funding, but there are scholarships and there are lots of other ways to get money. Medical students are great because they can get their own funding usually. It's tough. Finding the group that's already got success, already got some runs on the board, I think that's the way to go, particularly if you've got an interest in it to begin with.
How do you evaluate which opportunities to pursue and which ones to not pursue?
I think at the start of your career, you have to take every opportunity because it is what you make of it, I think. So, you know, at the start, say yes, 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 as much as you can. And then I guess after a while, you really have to think, you know, is this something I enjoy? Because really, you want to spend your time doing the things that you enjoy the most. What's going to make a difference? I don't know. I think you need to come back to those type of questions.
Yeah, that's all true. And I think, you know, I guess, where is this opportunity going to take me? You know, if I am successful, where is it actually going to lead to? Is it to a great extent a dead end, or has it got opportunity to then take either the research or the funding or whatever sort of further? So, it's tough, but I would agree, take every opportunity you're given, and certainly in the early stages.
I think also, particularly working with surgeons, gastroenterologists, a lot of them might want to just do the extra research to get that extra tick to go on to do whatever staff specialist. So I think also it depends what your reason for doing it in the first place is as well. Why are you actually going down this path? Do you want a whole career in research, or are you just looking to build some other aspect? So that also plays a big role.
Yeah, I guess it's tricky because you always got to consider you're already really busy, and you know your schedule's full, and it doesn't really change. you just get busier and busier. So taking on new opportunities means something else. You got to really evaluate where is it going to take you, and even though you say yes, particularly in early stage, you take all the opportunities you can, you still want to sort of try to think about where's that going to take you, where's that opportunity. So to have some high level, do you want to be a full-time researcher or a balanced academic, or you want to work in industry, or you're a clinician scientist and you want to, go on a particular pathway as well, and let that be the driver. You got to enjoy and work towards something rather than, pick everything.
I guess the other important thing, which Andrew alluded to earlier, is this is where a mentor comes in. If you have a mentor, these are the sort of questions you go and talk to them about and say, does this seem like a good opportunity, or should I be spending my time doing something else? And a good mentor will just talk you through. They won't probably tell you what to do, but they'll give you sort of pros and cons and sort of talk it through with you.
And I have one more question. I guess one of the things that a lot of researchers struggle with is maintaining and reaching a good work-life balance. And you are all extremely successful, so I wanted to ask you how hard was it for you to reach that? How important was towards your success, and what advice you can give in that direction?
Well, I'm happy to try that one. I guess I was just really pretty strong with myself that once I was home, I was home. And I almost invariably don't work at home. Sometimes I work very long days in the office, but I try very hard not to work at home. There are obviously exceptions where you just have to do things. But, really, and particularly when my kids were young, it's like I'm home and I'm here for my family. So it's really tough, but you sort of have to set your own ground rules and stick to them, and be prepared to say no when you have to.
Yeah, I'd agree with that. I completely ignore emails from Friday afternoon till Monday morning, and unless I'm working with the groups overseas, because they're awake when we're finishing. But even then, I'll ignore it till the morning. I almost never do anything at home, and haven't for quite a few years now. Because there is a life outside of this place.
Yeah, I agree with that. But it's really tough as you early career, because you're trying to get those opportunities, and if you're not working above and beyond, then on weekends and nights, then somebody else is doing it, and potentially they're more competitive. That's the academic culture, and I think that needs to change as people do exactly what Karen and it's important to say no and put these boundaries. And it's not always going to be possible, because sometimes a grant is due in 2 weeks, and you're going to be working day and night and weekends to get that grant in. But once you have that downtime, just try to strategically, make sure that you have time off.
I had a question, bit of change of tack. So we've heard a lot about supervision, grants, publications. So I want to talk about impact to consumers and community, and maybe just a reflection from each of you about the piece of work that you've done that you're most proud of, that has had the biggest impact on patients, consumers, and the community that you serve.
Yeah, I'm feel I'm still reasonably junior and to have a big impact in that space, but I do feel proud, again, that intersect that I had in experience working with industry, developing some of the sort of screening tools, which are consumer-facing tools for sleep disorders. And, I sort of didn't build the context that sleep disorders are really, really prevalent and major cost to health and community. And so having that tool, knowing that tool is out there, readily available globally, makes me feel like I achieved and contributed to helping with combat sleep disorders. And then working in health services around, developing guidelines and developing education for clinicians has been very rewarding, because that's a direct translation pipeline. And more recently is really working with consumers as consumer panels, actually talking to people with lived experience with whatever condition you're working on, has been an eye-opener, because what we think is important as researchers and clinicians, what we invest so much energy in researching and investigating, a lot of the time is not actually what what the consumer, what the patient sees as important to them, and that's been really important.
Yes, just to add to that, and probably relates to the question you asked, David Watch, our surgeon here, said to me when I first got here, we had this new technology. From when it's accepted and first published to when it makes any difference in, or changes practice or makes a difference, you're looking at about 10 years. And he's pretty much been spot on with that. So we've changed the way tests are done throughout the world, because once you publish something and you show what people have missed, they can't keep doing what they were doing. They have to change. What difference does it make? It takes a long time to gather that information to see what difference it takes. I mean, you can't change health policies quickly. You've really got to show that you've made a difference. So it's a long-term goal to do that. You can change a procedure, but what difference you're making in the community takes longer to sort of weave its way through.
Yeah, I think it's really important to, I mean, not everybody is necessarily absolutely sort of at the point of affecting the end user and the community, but it's really important that everybody in whatever project they're doing is working out what the final impact will be, and that there will be an impact and there will be benefit. I guess what I'm most proud of is the Medical Device Partnering Programme, which has worked with, as I said, probably we've done 120 different projects. Now, not all of those have now ended up on the market and affecting the consumer, but a reasonable number of them have. And certainly some of the things we've done is to ensure that ideas that are being developed and people who are putting loads of money in actually have had some clinical input to them, they've had some sort of manufacturing input to them, they've had input from all the various different stakeholders to know that it's actually an idea that's worth pursuing.
Yeah, just very briefly, I think, in the last few years, I've been involved in dementia guidelines, which has changed OT practice. We've brought out an evidence-based program from the US. We've trained hundreds of therapists. The actual practice of OT for dementia care has changed in the last few years, and we've done very translational work, and I think that's been a big step forward.
I've got two quick examples. One is some of the work that we did in the US. I worked as part of a quite large team on a virtual Iraq, virtual Afghanistan exposure therapy. So using virtual reality to implement an already existing therapy, which was the imaginal exposure therapy. We did a lot of work on the ground with consumers and then evaluating that tool, and it's now used in 40, now I think over 50 centres in the US to support veterans with post-traumatic stress. And more recently, the work that I've been got the most enjoyment out is I've been supporting a person with lived experience of stroke who was interested in undertaking her own research program, and so I've given her some training and been working really closely with her, and she's just completed a series of interviews with stroke survivors about what information is given to stroke survivors on transition out of hospital, and she presented that at a conference and just recently at the Smart Strokes conference and won an encouragement award. So that was really an exciting part of my career.
I've just got a question. So as a researcher myself, I've noticed some really changes in research, and particularly focus on implementation, translation, consumer engagement, things like that. As researchers and obviously very experienced in your fields, what have you noticed to be the biggest changes you've kind of seen in research, and what do you think, where do you think we're heading, and what do you think where might we be in, say, 10 years' time? Do you know? Does that make sense?
I mean, for me, it's all been about, big data, registries, AI, and that's been kind of the biggest thing I've seen, apart from those things you've already mentioned.
Yeah, absolutely. And certainly, I guess, in the technology field, that's absolutely, and, wearables and smart this and smart that and so on and so forth. Some of which is less smart, I have to say. I can't remember what the question was. What was the question? What's changed? There's absolutely, definitely more emphasis on, translation and consumer involvement and so on and so forth. I think still sometimes people are paying lip service to that, and I think there's still a way to go to actually really firmly embed that in research. But we're moving in the right direction.
I think the ability to share information and work with groups overseas has changed dramatically because it's so much easier now. One of the tests we do in patients, we surveyed 120 different countries around the world. All 120 did it a different way. Not one person did it the same way. We were able to pull together a group that set a guideline, and now everyone does it the same way, and that's the information that we're now pulling together. So it's the ease of being able to do that now, which we never could have done when I started. It was just too hard. And you didn't tend to share information either. You kept it to yourself. So at least in my world, that's changing quite a bit, particularly if you're working with paediatrics. They just will throw data at you because they're actually interested in trying to fix the kids. They don't care who owns it. So work with paediatrics, that's fantastic. Love to give you data.
I think it's became more like programs and large collaborative groups and consortia are real strength when you're going for large grants. It's difficult now being as a single or only a few investigators from a single lab getting up some significant programs. What was the other thing? And I guess, this whole translational pathway, working with industry is a lot more favourably looked at least in the last 5 years. When I was doing this and got involved in alertness CRC, it was all exciting at first, but then 2-3 years in, I suddenly realised, well, I'm not publishing anything, because you can't publish easily when you're working in a CRC. There's embargoes, there's IP. You get to learn about all that stuff, which is great, but then it's like, well, I need to publish. How am I going to be competitive for my next? And that was a real worry for me when I was going for my career development fellowship, because I didn't publish anywhere as much as I thought that I should be publishing if I wasn't involved in the industry. But the pitch and the application then is more around, well, there's a need for this translational research. There's a need and a push to work with industry. There's a need to get things out there and not just publications. And your investigator grants now, for example, are targeting that impact. It's not just about knowledge gain. It's about all those other elements as well. So I think it is shifting, and you can work comfortably with industry and work with clinicians collaboratively and not publish as much, but do all those other elements and be very successful.
Okay, I think it's time to wrap it up. So I just want to say on behalf of everybody to our wonderful speakers, thank you so much for coming out and and giving us all the advice and inspiration that we need. It's really fantastic and wonderful to see all the programs, all the networking, the collaborations, new technology, the diverse research areas. It's just fantastic to see. So I think we all enjoyed it thoroughly. Thank you all for coming out as well. It's late in the day, so that was really awesome. And I hope everyone has a really good weekend. Well, there's still another day left tomorrow, isn't there?